Good morning, ladies and gentlemen, and welcome to the first ICAVESS, or International Conference of Advanced Veterinary Science and Technology for Sustainable Development, organized by Faculty of Veterinary Medicine, Universitas Gadjah Mada, Indonesia. My name is Karisma Dewi, and it's been an honor for me to be here today as your MC for the next couple of days. As a start, I would like to greet Professor Panut Mulyono, Rector of Universitas Gadjah Mada, Indonesia. Good morning, Professor. Thank you so much for joining us. Professor Dr. Siti Isrina Octavia Salasia, the Dean of Faculty of Veterinary Medicine, Universitas Gadjah Mada, Indonesia. Good morning, Professor. Thank you so much for joining us.
Morning.
Professor.
Good morning. Thank you.
Thank you. Professor Masatoshi Hori, the President of the Asian Association of Veterinary Schools. Professor Michael Reichel, Professor Carlos Hermosilla, Assistant Professor Chan Pop Sawangmake, Professor Abdul Ahmad. Professor, thank you for joining us. our honorable guest, Professor Aris Junaidi, DVM, PhD. Good morning, Professor. Thank you for joining us. And all of the deans of the Faculty of Veterinary Medicine in Indonesia from Indonesian Association of Veterinary School. And of course, good morning to all of our participants. We would like to appreciate your presence despite your busy schedule. Ladies and gentlemen, as our next agenda, we'd like to hear the national anthem of Indonesia. So please welcome Indonesia Raya. Thank you. And the next one, ladies and gentlemen, to welcome you to this event, we'd like to hear the opening remarks. And first, I would like to invite Professor Dr. Siti Isrina Octavia Salasia as the Dean of Faculty of Veterinary Medicine, Universitas Gadjah Mada, Indonesia.
Assalamualaikum warahmatullahi wabarakatuh. Good morning. The Excellencies, Rector of Universitas Gadjah Mada, Professor Panut Mulyono, Professor Masatoshi Hori, the President of the ASEAN Association of Veterinary Schools from the University of Tokyo, Dr. Ronelo Abila, OIE Subregional Representative for Southeast Asia, Professor Carlos Hermon Sila from Justus Liebig Universität Gießen, Germany, Professor Michael Reichel from University of Hong Kong, Professor Cheng Pop Sawangmake from Chulalongkorn University, Dr. Sonia Kleinetz, DAAD Long-Term Lecturer, IBB University, Professor Her Bejo from University Putra Malaysia, Dr. Pandong Ryu from Seoul National University, and Professor Wayan Tunas Artama, Faculty of Veterinary Medicine, Universitas Gadjah Mada, the Honorable Invited Deans, ICAVESS members, presenters, and all participants. On behalf of the Dean of Faculty of Veterinary Medicine, Universitas Gadjah Mada, and the President of the Indonesian Association of Veterinary Schools, I would like to wish you a warm welcome to the first International Conference of Advanced Veterinary Science and Technologies for Sustainable Development 2021. It is truly an honor and a privilege for us to have the opportunity to speak and welcome you all to this wonderful occasion. Even though the world is still facing crisis due to disruption caused by COVID-19, We are grateful for organizing our first international veterinary conference in collaboration with the ASEAN Association of Veterinary Schools and having the opportunity to share it with you in a virtual way. The first ICAVESS 2021 offers the theme of synergizing sciences for sustainable animal, human, and environmental qualities in global change. This conference will share the recent advances in veterinary science and technology and concern the global climate change and anthropogenic factors, which unavoidably lead to significant disease distribution worldwide. In this event, we invited a prominent scientists in their respective field from Japan, Germany, USA, Thailand, Malaysia, Korea, and Indonesia, as well the OIE subregional representatives for Southeast Asia to share their experience, research, and opinions on the global change of climate and diseases. Today's conference also certainly marks a special occasion for veterinary school deans, an opportunity to discuss and share information, possible collaboration in educational improvement in terms of the standardization system, globalization, and challenges to veterinary education in Asia. I am sure that networking and collaboration will be established among us, strengthening the future excellent veterinary education, research, and technology for Indonesia and the world. I would like to express my sincere appreciation to the committee who have made the conference runs even in virtual way. This is the first ICAVESS conference that is conducted on a digital platform in line with the social distancing norms due to the COVID-19 pandemic. We sincerely hope that the symposium and all related activities will also produce a positive impact and benefit for veterinary education, research, and possible synergizing sciences for the sustainable animal, human, and environmental qualities in the world. Thank you for joining the conference. I do hope you will also take the time to enjoy this conference. Thank you. May Allah bless all of us. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you so much, Professor Isrina, for your warm welcome. Up next, we are going to hear the opening remarks by Professor Masatoshi Hori, the president of ICAVESS. So ladies and gentlemen, please welcome Professor Masatoshi Hori.
I'm Masatoshi Hori, head of the Department of Veterinary Medical Science, the University of Tokyo. And also, I'm a president of ICAVESS. Congratulations on hosting the first international conference of advanced veterinary science and technology for sustainable development, ICAVESS. So I would like to express our deepest gratitude to the Dean of the Faculty of Veterinary Medicine, Gajah Mada University, and also the organizing committee in this meeting. So due to the COVID-19 pandemic, the event was postponed last summer. This time, it will be held online. But the fact that ICAVESS will be hosted is due to the effort of committee members involved. Thank you again. By the way, so I myself have never visited Indonesia and was looking forward to visiting Indonesia because my wife, a child for 6 or 7 years, she often told me about Indonesia's greatness, especially nature and foods for me. So this time, I will be participating by online system, but I will definitely like to visit Indonesia and Gajah Mada University. Dean Sato, Dr. Koichi Sato of Yamaguchi University, he's my longtime friend. So I'm thinking of having him escort me. By the way, the role that veterinarians should play in the world is increasing year by year. There is an urgent need for veterinary education, veterinary research, and veterinary human resource development in Asia, such as infectious disease problem, food safety, food security, and antibacterial drug-resistant bacteria. At the United Nations Summit in September 2015, the 2030 Agenda for Sustainable Development was unanimously adopted by the member states. So it is Sustainable Development Goals, SDGs, our international goal for the sustainable and better planet by 2030. SDGs are international goal for sustainable. So SDGs are important for global conservation, including humankind. I have high hopes that this ICAVESS meeting will become one of the core and contribute to the SDGs in the future. So finally, in the last 1 year and 4 months, many people around the world have died of SARS-CoV-2 infection. I would like to conclude my greeting with my condolences.
Thank you, Professor. And ladies and gentlemen, we'd like to hear the opening remarks by the Rector of Universitas Gadjah Mada. So please welcome Professor Panut Mulyono.
Assalamualaikum warahmatullahi wabarakatuh. May peace be upon all of you. Dear distinguished speakers, participants, ladies and gentlemen, on behalf of Universitas Gadjah Mada, I warmly welcome all of you today at the first International Conference of Advanced Veterinary Science and Technologies for Sustainable Development 2021, organized Gadjah Mada. I would like to take this opportunity to congratulate the Faculty of Veterinary Medicine UGM and the Asian Association of Veterinary Schools for organizing this international conference online. I would also like to extend my gratitude to the keynote speakers, Professor Masatoshi Hori from the University of Tokyo, Dr. Ronelo Abila, OIE, Sub-Regional Representative for Southeast Asia, Professor Carlos Hermosilla from Justus Liebig Universität Gießen, Germany, Professor Michael Reichel from University of Hong Kong, Professor Chen Pop Sawang Maki from Chulalongkorn University, Dr. Sonia Kleinert, DAAD Long-Term Lecturer, IPB University, Professor Khair Bejo from University Putra, Malaysia, Dr. Pan Dong Ryu from Seoul National University Artama, Faculty of Veterinary Medicine, UGM. As a brief introduction, Universitas Gadjah Mada is the oldest and the largest university in Indonesia and considered one of the best universities in Indonesia in 2020, according to the Ministry of Education and Culture of the Republic of Indonesia. Since its early establishment, UGM has been the place to learn and grow for the young generation of Police campus. UGM creates a conducive environment for the continuity of the learning process. It is our way to develop multidisciplinary collaboration and respond to ecological issues to achieve the university's vision. UGM also promotes ecological development by restricting the number of vehicles entering the campus, constructing green dormitories, and reducing pollution. Of the COVID-19 pandemic, UGM is working hard multidisciplinary research and innovation such as the genus is necessary for our present and future. Preventing environmental degradation is mandatory for us, for our succeeding generation. The first ICAVESS concerned the global climate change and anthropogenic factors that lead the significant change in disease distribution worldwide, including COVID-19. Multidisciplinary studies act as both response and a means to develop sustainable strategies that can function across continent and administrative boundaries. Sustainable development inspire ICAVESS as the development that meets the present needs without compromising future generation ability to meet their own needs. Thus, the general theme for the ICAVESS 2021 is synergizing sciences for sustainable animal, human, and environmental qualities in global change. We hope ICAVESS will inspire other academic or researchers to bring an even more significant impact in life science. UGM is proud to lead the way in facilitating cutting-edge information dissemination from interdisciplinary research and is pleased to conduct this virtual conference. Hopefully, it can strengthen our collaboration, especially in veterinary, life sciences, and the environment. Finally, I wish you a pleasant and enjoyable conference. Thank you very much for your attention.
Thank you so much, Professor Panut. Ladies and gentlemen, this wonderful dance you are about to enjoy is called Klono Topeng. Klono is the name of the king from Puda Payung Kingdom, and Topeng means mask. Originated from Yogyakarta, this beautiful dance depicts a king who fell in love with goddess Sekataji. And with this, even though virtually, we also hope that you will fall in love with our beloved city, Yogyakarta. So ladies and gentlemen, please welcome Klono Topeng.
Tarian ini adalah tarian klono topeng gaya Yogyakarta. Tarian ini menggambarkan tokoh besar yang bernama Prabu Klono Sewandono yang berasal dari kerajaan Pudak Payung yang sedang jatuh cinta dengan putri kerajaan yang bernama Dewi Skartaji.
What a wonderful dance. Ladies and gentlemen, as we all know, COVID-19 created a huge impact on every sphere of human life. It is possible to explain the negative and positive impacts of COVID-19. Despite the big loss for economy, COVID-19 also strengthening healthcare infrastructure and research. Moreover, it also raises many small-scale industries. Last but not least, the use of digital tools and virtual meetings like this are inevitable in this new normal era. Ladies and gentlemen, the sustainability of development are needed to present it for future generations. Therefore, ICAVESS proudly presents Synergizing Science for Sustainable Animal, Human, and Environmental Qualities in Global Change. The first ICAVESS also concerns about the global change of climate and anthropogenic factors, which are unavoidably lead to significant changes in diseases distribution worldwide. The collaboration of multidisciplinary studies is needed, not only as a responsive reaction, but also to develop a sustainable strategy that may cross the boundaries. Professor Michael Reichel, Professor Carlos Hermosilla, and Assistant Professor Chen Bob Sawang Make will be our keynote speakers of today's plenary session that will be led by Professor Teguh Budipitoyo. And after that, it would be followed by the Asian Association of Veterinary Schools on Vet Education System that will be led by Professor Omar. And last but not least, we're going to have the parallel session in the end of our meeting today. And ladies and gentlemen, you may see on your screen our schedule of today. We can see that there are Professor Michael Reichel, Professor Carlos Hermosilla, and also Professor Chen Pop with a great topics for today's. And after that, we're going to have some rules since this is virtual meeting. So I hope that you are going to understand of our conditions. And you may see it on your screen. And of course, please mute your microphone during the plenary session, unless Professor Teguh led you to do that. And also, we also want to remind you to rename your account name with the following format: name of the presenter dash your question and your email. Oh, I'm sorry. I mean, this is the format for your questions. Okay. Last but not least, ladies and gentlemen, our organizing committee will provide it with the attendance form. So don't forget to fill out the attendance form that you can see in the chat box. And don't forget, you have to attend the full session from the beginning until the end to earn the certificate. Ladies and gentlemen, let me introduce to you Professor Teguh Budipitoyo as our moderator for this session. Professor Teguh Budipitoyo is a lecturer and a researcher at the Department of Anatomy and Faculty of Veterinary Medicine, Universitas Gadjah Mada, Indonesia. Nowadays, he serves as a vice dean in our beloved faculty. So without further ado, please welcome Professor Teguh Budipitoyo. Professor Teguh, may I ask you to unmute the microphone, please?
Okay.
There you go. Thank you, Professor.
Thank you for the brief introduction, Karisma. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Once again, welcome to the first International Conference of Advanced Veterinary Science and Technologies for Sustainable Development 2021. First, let me introduce myself. My name is Teguh Budi Pitojo, and today I will be responsible as the moderator for this plenary session 1. I'm glad to inform you that we will have 3 wonderful invited speakers for this session: Prof. Michael Reisha, Prof. Carlos Hermosilla, and Assistant Professor Dr. Chen Pop Sawangmake. Each presentation will be delivered using pre-recorded video for approximately 30 minutes, followed by 10-minute discussion. During presentation, if you have any question, you can use the chat box to deliver your question according to the format that will be informed by the organizer via chat box. The first presentation of this plenary session will be given by Professor Michael Reisha. He is recognized as a specialist in veterinary public health in Hong Kong, Australia, and also New Zealand, as well as in Europe. So the organizer, please, you can play the video.
Thank you and good morning from Australia. In the current talk, I would like to explore the global impact of, in particular, the current pandemic of COVID-19, and its impact that it's had on us in Australia, I'm sure in Indonesia and other parts of the world, but also the opportunities and challenges that this presents for us, not only for the current pandemic, but also for us looking forward into the future. It's only just over a year ago that we first saw the reports emerging out of China of a coronavirus-like disease that then started to envelop the whole globe. So at that time, the whole epidemic had only just logged on just over 5,000 cases. These were mostly centered in the Hubei province, Wuhan in China, and there's been very few dots around the world. From those initial 5,000 cases that we saw early on, we've now gone to 120 million cases globally as of March 2021. There are, if you believe this graph, encouraging signs that the current epidemic is decreasing, so the number of daily cases is seemingly reducing, and by quite a large margin. There's also encouraging signs that the death toll that we have seen is reducing as well. So the total death toll, again, as of yesterday, was 2.6 million, which is probably about 2% on the total recorded globally. It would be nice to think that this is related to the COVID vaccine rollout, which is seemingly progressing quite well. As you can see, there are a number of countries, particularly Israel, that seem to have delivered at least one dose to every one of their citizens. And if you recall that 2 are required, they obviously have a fair way to go yet, but others are catching up quickly. United Arab Emirates, in the UK, and other countries, Chile in particular, they're all catching up with the vaccination game. There are encouraging signs with the number of new cases and hospital admissions and new deaths being recorded here in the UK. that this seems to be working. So ever since they started vaccinating at the beginning of December, those numbers have come down quite considerably in the age groups that have been vaccinated. Well, did it all start in the market? I mean, this is obviously still subject of a WHO investigation. I mean, there's certainly very strong indications that a market was somewhere in the middle of it. I mean, maybe as a further amplifying, venue, but there certainly seems to be a zoonotic angle to this pandemic. The strong indications that we have bats involved is the host of the COVID-19 coronavirus. With something like 91% similarity to other known strains of coronaviruses. That it makes a lot of sense. It's similar to the story that we have with SARS and MERS, and we talk about this a bit later on. And there's some indication that this probably diverged about 50 years ago, so it's been around for a while. Whether it's been around in the pangolin as an intermediate host, it's another question, but it's been around, and it's just decided to pop up, as the circumstances, were ripe to, move into the human population. We're not talking just about bats and pangolins potentially, but there was early indications that other species of animals were infected, or infectable, like dogs in Hong Kong. That was one of the early reports. There were cats in the Bronx, wild felids, tigers in the Bronx Zoo, but also cats, domesticated cats in Wuhan, which showed a very strong, and high antibody presence after the outbreak occurred there in January, March. And minks, which have shown signs of increased mortality, and large numbers of them have been destroyed. Experimentally, we know that other species are susceptible: hamster, ferrets, monkeys, but not chickens, pigs, and ducks. The biggest in terms of animals calamity has probably been visited on minks. I mean, there's been a large number, millions culled in Holland and Spain, and also I believe Denmark, Scandinavia, where the industry, particularly in Holland, has brought forward, was closed down, and that was brought forward from originally scheduled to be 2024 to 2021 because there was a large number of cases here, close to 100 cases where people living or working on mink farms were diagnosed with COVID-19, and that seems to be passed on between humans and humans, and humans to animals, and back from animals to humans. And I mean, there's the cost of it all. These estimates back there from January last year. I mean, obviously highly optimistic. At that stage, Australia was thought to maybe be in for $1 billion Australian, but if it doesn't get worse, I think is the rider on this. And when we're thinking that the net debt of Australia is doubling now to $1 trillion. So there's probably out by the factor of 500. The global economy is also estimated to be there to be losing 1.1 trillion. But even the last help subsidy package in the United States was already worth $2 trillion. So I think we are underestimating these costs by a huge margin. This was a side of the previous estimates of some of these pandemics that have been visited upon the globe in the last 25 odd years, and you can see here SARS was a big one with 50 billion. The avian flus and flu pandemics, Ebola there was $10 billion, but this is all dwarfed by probably the trillions of dollars that have been spent on either combating COVID-19 around the world or aiding the recovery from it. So where do we come in as veterinarians? What can we contribute? What we have contributed are sort of ventilators. So there was certainly early on in the crisis call for ventilators, which originally coming mostly from the human model design anyway. And there was a survey done that suggested that maybe up to 10% in addition of ventilators could be sourced from veterinary clinics. The biggest bottleneck, however, was trained staff, and the authors in this article, even contemplated drawing on veterinary nurses, but not too sure how well that would have gone down in the medical fraternity or medical hospitals. But albeit a possibility. I don't think anything has really come from that, because critical shortages weren't ever as critical that they had to, in their opinion, stoop as low as borrowing equipment from the veterinary hospitals. Early on, there were also reports from people, particularly I think originally in France, but also in Australia here and in Germany, from folks training dogs to identify by sniffing COVID-infected people. The tests were remarkably sensitive, and I mean the nice thing about dogs is that they can do the tests in seconds. Only some cases took a week to be trained. And I understand that Dr. Carter was actually involved in the development of, call it an AI dog, a sniffer device which could actually do this electronically. So, I mean, there were some contribution. I'm not too sure how much of this has actually been used, but there's a picture of this being done in Helsinki. And I mean, even as we are moving out of the pandemic and we're trying to open up borders again, I mean, this might be something that we shouldn't forget about. This is where a contribution could be made to enable international travel by plane, by checking people before they got on the plane, before they embark on their journey. At Cornell University, the Animal Health Diagnostic Center set up a purpose-built COVID testing lab within the first 50 days, and that was certified by the health authorities, and was able to test anything up to 7,000 students and faculty per day, not all individually, but in pooled samples. And they were able to develop a system that allowed the university to track how they were traveling with COVID-19 infections, and develop this traffic light type scenario where green was all good, the new normal, and red was obviously something that was of greater concern. But this was an opportunity for veterinarians to make an impact, as have others in veterinary epidemiology and the molecular sciences, as the case may be in the various countries. The important thing, though, is that the population perceives that what you do is being coherent and confidence inspiring, and it needs to be done, from the same song sheet. So I mean, this has been a slight issue in Australia, where we had different states doing different things for a while, but I mean, in a sense, most people have actually now started to believe, that governance is science-led, and that we're all in it together, and we're all travelling in the right direction. The direction needs to be embarked on very quickly. So, I mean, early closure of borders, early action on new clusters is very important. And as Dr. Ryan here says in his quote, I mean, the greatest error is not to move. So if you're paralyzed by the fear of doing something wrong, you're never going to be able to catch up with the virus, because it moves so fast. This level of confidence in the trustworthiness and the believability of our decision makers, particularly here in Australia, has been quite important in establishing the rapport with the population, supporting the evidence from trustworthy scientists, whether they be medical or, in our case, maybe veterinary, and then support of these experts through the governing politicians has been instrumental in making sure that, for instance, in Australia, for the first time in a year, we've got all the borders open. There's no impediment to travel within Australia, although we still have the borders closed and trying to maintain, the disease, outside of our borders and not letting it in. For the most part, the current pandemic has been and will be a public health emergency. It will be led by medicos. It will be, hopefully supported by veterinarians. And I mean, there may well be, as I said, for instance, in the case of the sniffer dogs, be an opportunity to still play a part of it, as we're moving out of the pandemic again. But looking forward into the future, I think it is important to continue to strengthen our connections with, the other medical sciences, the medical fraternity, and strengthen those in peacetime before the next disease occurs. So, we need to look at jointly at disease prevention, recognition, management, and elimination of diseases before they become a problem. And this is what I want to spend the rest of my talk on discussing. Here I want to talk about the notion that we should be sort of worried, about wildlife. We should be concerning ourselves with wildlife, because the analysis of emerging infectious diseases over the last 75 years, 334 EIDs analysed, since 1940 suggests that 72% of them originate from wildlife. And I mean, I'm just mentioning a few examples here: SARS, Ebola, MERS. They all, originate from wildlife. And in Australia here, we've seen, the emergence of diseases that were previously unknown and unrecognised, such as Hendra, Australian Bednester virus disease, and Meningel virus, and so on. When you look at this map here, then you see that, I mean, obviously our region, from the subcontinent, India, Bangladesh and Pakistan, across China, the Southeast Asia, and down to Indonesia. I mean, this is the area where, the relative risk of an EAD emerging and, causing another problem is the greatest. Why is this happening? So when you look at the animal-human interface, I mean, this has in the past been a relatively, well-ordered sort of interface where there was occasional, interaction and communication between the animal populations, moving out into foraging, areas, the agricultural land, and humans being, involved in the forest. But I mean, new with deforestation and landscape fragmentation, these opportunities for interaction and contact between those two populations has increased. And if the last slide, you can see that with these multiple bubbles of smaller areas being left over for wildlife, there's a lot more opportunity for human-animal interactions, and with that also an exchange of sort of zoonotic agents. Some of these, and they happen to be all bat-related. So when things like Ebola, which has emerged, with strong outbreaks in 2014, 16, 18, and 19, and again now in West and Central Africa. SARS, some 20 odd years almost ago in China and Hong Kong. The henipaviruses, Hendra and Nipah, in about 25 years ago, and 20 years ago, and MERS and MERS coronavirus in the Middle East and Korea. Initially, when you look at the history of Ebola, nobody really cared because it was these small outbreaks. They've been going on since 1976. But then in 2014, we had the big one, and everybody started to get worried. The current outbreak, involves something like more than 3,000 cases, but previously the 2014-16 outbreak involved close to 30,000, and 11,000 of them died, so a high case fatality. The ecology, obviously, as we said, it involves bats at the center of it. I mean, they sort of pass it on to non-human primates and wildlife, which may have contact with human populations, and you have spillover events where they pass it on then to human populations, which then in turn pass it on to other humans. So then you get human-to-human transmission, and whether this is funeral practices, health workers, or blood and body fluids, just contact, through the caring for infected people. All of those are possibilities in, passing this on to other populations. I mean, this is a simplified diagram of, how you can imagine this happening. So you have, the wildlife animal cases, ticking over in waves at the bottom of the screen, but then with spillover events, you get other populations, maybe domesticated animals such as cows and sheep and goats and poultry involved, or pigs. And then, this again, these might act as amplifier hosts where they're actually increasing the titers of viremia of viruses, and this spill over into human populations, which might amplify it in themselves or just continue to be infected from these spillover events. If we're looking at the SARS and MERS, another two coronaviruses which we've had experience with globally, the 2002-2003 outbreak of SARS led to more than 8,000 infections worldwide, mostly in China and Hong Kong. We killed about 800 of them, so a case fatality rate of about 10%. That was a coronavirus that made the species jump from bats to humans. through the intermediate host of farm civet cats, and that was a story that took us, probably near enough 10 years to unravel, and it's not seen ever since 2004. So when this has been gone and unfortunately maybe a little bit forgotten about. But the Middle East and respiratory syndrome MERS outbreak has been recognized since 2012. It's been fewer than about 3,000 cases of MERS and in the world, and with limited spread outside of the Arabian Peninsula. So it's been a very much of a regional thing, with a albeit high case fatality rate, which infections primarily affecting older individuals with comorbidities, and killing about 40% of them. So we have the two other coronaviruses there that affected humans. You've got SARS at the top, so bat populations, the intermediate host, the palm civet, the cats, these farmed animals that have occasionally, in rare events, passed it on to humans, zoonotic transmission. Whereas MERS, over a number of years, and on a continuous basis, have been passing it on through probably the intermediate host of dromedaries to human populations. And then, once it's in human populations, you have human-to-human transmissions in the hospital setting, through healthcare workers or other patients, and so it continues. Now I want to switch continent and actually talk about a recent study here in Adelaide, and it just is an example of how you can proactively, start to look at what's actually going on in the neighbourhood. So grey-headed foxes, flying foxes, have not been in Adelaide until 2010-11, and the population that arrived and set up camp there right in the middle of basically central Adelaide, grew from 300 back in 2010 to now over 20,000. Fecal analysis suggests that they might actually be attracted by introduced flora. So they like what we provide with them, and so it makes a lot of sense, and it does give you understanding on why they actually come here. So originally they were only on the east coast and the northern parts of Australia, but they're now starting to make their round, around the continent to the southern parts of Australia. And when you look at them, and these are the samples from antibody serology of 301 of them, you can see that there's antibodies for SARS, and something like 40% of those are suggested to be infected or having signs of having previously been exposed to SARS, and a very few, only 1 and 2% for MERS. But the other one is Hendra virus, which we know has affected bats, and that seems to be prevalent all around Australia. So we have a very nice distribution of unaffected Hendra bats on the left, and something like a 20%, 26% population on the right. There's no evidence in this particular survey of antibodies to Australian bat lyssavirus, although we know that a bat died in 2012 in Adelaide from rabies or Australian bat lyssavirus. So it does occur, but it does seem to be a relatively rare. Talking about Australian bat lyssavirus, we know that it's very closely related to classical rabies. We've had 3 human deaths in Australia. The infection is estimated to be very low in healthy bats, which does agree with the survey that's presented on the previous slide, so very low prevalence. But when they get sick, or when they are sick and injured, there's a much greater chance of them being infected with ABRV, and this is why we recommend that all veterinarians, nurses, and health wildlife carers and students in Australia are vaccinated for rabies. Hendra, out of the henipaviruses, has also only been recognised in Australia, but recognised for the first time in 1994 in Hendra in Queensland, near a suburb of Brisbane. It was transmitted to horses from fruit bats with human infection as a result of direct contact with infected horses, which seem to act as an amplifying host. They themselves get it from bats, but the case fatality rates in both horses and people is very high. So, out of the 7 infected humans, 4 of them have died, 57%, and 2 of them are veterinarians, which again demonstrates that we are in the firing line, and we are the first ones to be potentially exposed to something new. So we should keep our wits about us and be aware of the possibility. This is the distribution. You see there in pink, Hendra virus in Australia and maybe up into Papua New Guinea. This is, I think, hypothesized. It hasn't been diagnosed, but Nipah in India, Bangladesh, and the Philippines. Basically, we've got different possibilities of looking at managing the exposure. We can look at the local bat density and the abundance of bat food in trees. And I refer back to my earlier mention of introduced species making it particularly attractive for bats to visit. The husbandry, so certainly in Queensland, it's been suggested that you don't raise horses under mango trees anymore, which seem to be a favourite food of bats. And we're also looking at horse susceptibility, and here particular acquired immunity, which is the vaccine that I'll be talking about later on. The disease is influenza-like, respiratory, but also neurological signs, and also the indications of relapses. So there's been one human case that relapsed a year later and then ultimately died. And that has also been reported for Nipah virus. Horses die after a very high fever. So you see in this, in the top slide, under A, that they all, get temperatures up to 39, 40, 41 degrees, and then they die. But they shed the virus potentially from day 2. So any horse in the northern parts of Australia can be sort of potentially a Hendra case. Again, when bats seem to be not infected clinically, so they seem to be shedding at a low rate, maybe 2.5% of bats, but they don't show any disease. We were looking at Nipah, the first outbreak. Pigs is being relatively mildly affected, but acting as an amplifier host for humans, which then after an incubation to 5 to 18 days, it sort of became sick. There's been suggestions that you can also get affected from drinking raw palm sap as the most common pathway, which has obviously been suggested to be infected by maybe urine contamination from bats or their saliva. And there's also been the suggestion that undercooked or meat preparation, the slaughter and the cutting up of sort of Hendra and Nipah infected horses might have been contributing to an outbreak in the Philippines. We're talking about similar viruses, but different behaviours or different outcomes. So we have at the top, we have Hendra in Australia and Nipah in the Philippines with very high mortality, facilitated through a transmission cycle from bats to horses to humans. We've got pigs involved with Nipah in Malaysia and Singapore, and human-to-human transmission in Bangladesh and India. We've been fortunate enough to have a vaccine for Hendra now since 2012 in Australia, which does seem to induce very high antibody levels, which last for up to 12 months after you had the first series of 3 immunizations. The problem is it's quite expensive, so we're talking about $200 for each of those vaccinations. And but when they do get vaccinated appropriately, then they seem to be resistant to subsequent infection. It would be nice to think that you see here the event timeline of Hendra infections or events in Australia. When we had a sort of a burst of activity with 18 events in 2011, but this has come down, and the release of the vaccine would seem to be timed with that very high incidence of Hendra occurrences. But it would be nice to think that with the advent of the vaccine, these numbers have come down, and we're now talking again about 1s and 2s for every one of those years from 2018 onwards. Obviously, in the ideal world, all horses should be vaccinated, as humans should be for COVID-19. But the reality is that, in particular, with an expensive vaccine as it is, only decided to vaccinate their horses. Just to be, although bats seem to be a particular problem, and when there's believes there's about 1,400 different species of bats, and there's certainly been a lot of viruses isolated from them. But here's an example of a bacterial disease, Brucella surs, in pig hunting dogs in Australia, which is, we've known about this for a while. It's been generally regarded as a problem in Queensland, southwest Queensland, sort of inland from Brisbane, but it's now spreading, being recognised in other parts of, particularly northern parts of New South Wales. And it's obviously a disease that not only affects dogs, but it's also zoonosis, so it is potentially quite devastating. And there have been 2 human cases in Australia who luckily got away reasonably well. But I mean, it's potentially quite a serious disease as humans that we should be aware of. So in summary, this is a complex and changing situation and will continue to be so, and will challenge us, but we should also be always expecting that there might be something new out there and we should be looking out for it. That's why I think wildlife surveillance is quite important. It's not only just looking at it when the things have fallen off the rail, but it's looking like the Adelaide study shows, being preemptive and being aware of what is happening in your neighbourhood, and what these new arrivals, these new immigrants, such as these bats in Adelaide, central Adelaide, might bring with them. It's something we should be expecting something more and something different all the time. I mean, we know that there are things that we probably don't know yet in bats. As I said, there are plenty of isolation of viruses that haven't got a disease to their name yet, but there's potential there. And I think the last thing I would like to stress is that we should be thinking of it as something that concerns us in particular, because we're the most likely ones to be called in to look at a moribund bat or moribund pig or whatever the case may be, something that is of animal origin, and be asked to sort of make it a diagnosis. And so we should be sort of thinking about being safe, being aware that this might be a zoonotic disease, and getting there with the appropriate personal protective equipment, and if potentially there's vaccines available, such as the rabies vaccine, we should be using this as a preventive measure and not just be thinking about it after the fact. I'd like to thank you for your attendance and for your interest in this talk. I hope it's been of interest. I mean, this is, as I said, an involving story. My previous, and this is the picture here, my previous appointment was in Hong Kong, where I was the foundation dean of a veterinary school there, which is obviously trying to make a contribution in part of the world by providing high level and research. And I hope that we can continue to do this, and I thank you again, and I wish you a good day. Thank you. Bye.
Thank you very much, Prof. Rachel. I hope you all enjoyed this amazing presentation. Global impact of infectious disease challenge and opportunities. Dear ladies and gentlemen, we understand that you are so interested to have a live discussion with Professor Rachel, because of his presentation is related with present condition, I think, and how one health approach is very important to solve the problem. But unfortunately, due to his tight schedule, Professor Rachel cannot join with us today. So the organizer will collect all of your questions, and Professor Rachel will respond and give answer to you by email. Ladies and gentlemen, now we will have 15-minute session break. Please enjoy video profile and student activity of Faculty of Veterinary Medicine of Universitas Gadjah Mada, which has prepared by the organizer. Before we move to the next invited speaker presentation. Please, the organizer.
Universitas Gadjah Mada is an Indonesian public Ivy League research university and is the oldest and largest institution of higher education in Indonesia. UGM has been considered to be one of the most prestigious universities in Indonesia. Faculty of Veterinary Medicine UGM has been developing knowledge and science since the early establishment of the university back in 1946. This started since it was still unified with Bogor Veterinary Medicine College, until a parallel class was opened in Klaten in 1947, which became the pioneer and forerunner of Faculty of Veterinary Medicine UGM. Currently, Faculty of Veterinary Medicine has 4 programs: Veterinary Medicine Study Program, Veterinary Profession Education Study Program, Master of Veterinary Science Study Program, and Doctor of Veterinary Science Study Program.
Fakultas Kedokteran Hewan Universitas Gadjah Mada lahir sebagai bagian perjuangan Negara Kesatuan Republik Indonesia yang memiliki cita-cita mencerdaskan kehidupan bangsa sekaligus berperan dalam perdamaian dunia. Maka Fakultas Kedokteran Hewan UGM memiliki visi menjadi pusat unggulan pendidikan tinggi kedokteran hewan bertaraf internasional yang dilandasi jiwa Pancasila. Visi tersebut diwujudkan dalam proses pendidikan yang tetap menjunjung tinggi kearifan lokal, namun tetap mengikuti perkembangan teknologi kedokteran hewan secara global di era revolusi industri saat ini. Fakultas Kedokteran Hewan UGM mengembangkan riset dengan memanfaatkan kemajuan teknologi, menghasilkan karya inovatif yang berkualitas tinggi, sekaligus aplikatif dan tetap berkarakter kerakyatan, sehingga dapat diadopsi dan dimanfaatkan oleh masyarakat secara luas. Sumber daya manusia yang berkualitas tinggi didukung fasilitas yang baik menjadi keunggulan Fakultas Kedokteran Hewan UGM mewujudkan insan kesehatan hewan yang unggul di berbagai bidang seperti di bidang laboratorium diagnostik, entrepreneur, praktisi, satwa liar, ternak produktif, penyidikan zoonosis, penjaminan keamanan dan kehalalan produk hewan, hingga mengambil kebijakan dan keputusan di bidang kesehatan hewan. Demikianlah peran Fakultas Kedokteran Hewan UGM mewujudkan visinya yang mendasarkan profesi dokter hewan pada filosofi manusia meriga satwa sewaka, mensejahterakan manusia melalui kesehatan hewan.
Veterinarians are expected to take care of animal welfare, starting from providing food supply, quarantine, reproduction, treatment, until controlling the spread of contagious animal disease. Various tasks and expertises of veterinarian are learned in 12 departments of Faculty of Veterinary Medicine UGM. Department of Anatomy is one of the oldest departments in the Faculty of Veterinary Medicine UGM. The main lecturers in this department is the Principal of Animal Anatomy. Department of Anatomy has two laboratories, Macroanatomy and Microanatomy. Department of Biochemistry develops research related to genetic diversity. This department also focuses on the development of the bird sexing based on DNA structures. Equipped with an advanced laboratory, the Department of Pharmacology is responsible for drug science, including pharmacotherapy and chemotherapy on animals, drug prescription, and research on drug effects on animals. This department also developed veterinary pharmacy, science in drug supply and drug management. Department of Physiology teach about the science of an animal's normal body function. The greatest research of Department of Physiology is the physiology response from various animals. Various development has been done in this laboratory, such as hormone testing and research on animal digestive nerves and development on stem cells. Department of Surgery and Radiology handles animal disease using advanced equipments in the veterinary medicine field, such as X-rays and USG. This department is facilitated with a special clinic for skill development and also as the community service unit. Department of Internal Medicine teaches about the clinical and laboratory examination techniques. The greatest research in the Department of Internal Medicine is the development of diagnosis and therapy for various veterinary internal medicine using advanced technology. Department of Veterinary Public Health plays a role in science of supplying animal source foods that are safe, healthy, intact, and halal, and also plays a role in the research of controlling infectious disease that can be naturally transmitted between animals and humans or zoonosis. Department of Microbiology develops science related to bacteria, virus, and fungus. This department plays a role in controlling important animal disease such as bird flu, rabies, anthrax, and SARS. Department of Parasitology develops research related to parasites such as worms and protozoa. This science plays an important role in controlling animal disease caused by parasites. The greatest research in the Department of Parasitology is vaccine production and molecular biological study on parasites. Department of Pathology has a laboratory that studies about cancer, diagnostic development on poultry and aquatic animals disease. Department of Clinical Pathology studies about animal disease diagnosis, drug side effects, and chemical materials. This department also develops research using animals based on proper science standards and animal welfare. Facilitated with an obstetric and gynecological laboratory, Department of Reproduction develops animal reproduction science, including reproductive disorders and reproduction technology. Various sustainable courses are held by this department, such as the inseminator and sterility courses. Besides advanced laboratories inside all of the departments, Faculty of Veterinary Medicine UGM is also supported by Animal Health Education and Training Unit or UP2KH. Until now, UP2KH managed several livestock such as cows, sheep, and horses. The facility in this unit is not only used by university students but also by veterinarians for the sustainable learning process. The learning process in the faculty always devoted by science and competencies to try to fulfill the society need. One of these is through research developed by the teaching staff, the discovery of the vegan chicken carcass testing kit that is fast and accurate. This testing kit has been patented and has widely used in the industrial scale, laboratory, and household scale. Faculty of Veterinary Medicine lecturers regularly write and publish books related to the veterinary medicine world that can be used for readers in the animal husbandry, biology, and statistics. The conducive learning atmosphere is proven effectively supports the university students to obtain various achievements, especially in the research field, arts, sports, and national and international organizations.
Dalam studi saya di fakultas ini, saya memiliki minat dalam bidang penelitian ataupun riset berbasis kedokteran hewan. Dan didukung penuh oleh Fakultas Kedokteran Hewan, saya mampu mengembangkan diri dan berprestasi selama belajar di sini. Melalui program kreativitas mahasiswa, atas bantuan dosen pendamping yang berkompeten, saya dan tim mampu lolos dan berhasil mengerjakan 12 proposal program kreativitas mahasiswa bidang penelitian. Mari terus berprestasi mahasiswa FKH UGM, manusia meriga satwa sewaka.
Halo, nama saya Ande Firmansyah, mahasiswa Fakultas Kedokteran Hewan Universitas Gadjah Mada. Selain aktif di organisasi lingkup fakultas, saya juga dipercaya menjadi pengurus inti dari organisasi mahasiswa kedokteran hewan global atau International Veterinary Student Association. Menurut saya, unit kegiatan mahasiswa yang ada di FKH UGM cukup banyak. Meskipun begitu, saya berterima kasih kepada FKH UGM karena telah membantu kami untuk menyalurkan minat, bakat, dan potensi diri kami. Besar harapan saya, dengan adanya bantuan ini, mahasiswa semakin aktif dan berprestasi. Manusia meriga satwa sewaka. Viva Veteriner Indonesia.
For the sake of developing the veterinary medicine science, the faculty established relations with national and international stakeholders. Faculty of Veterinary Medicine initiated the Veterinarian Medicine Association Indonesia through an MOU between the government and private sectors for research and community service. We also have collaborations with foreign FVM universities from Australia, Thailand, and Japan for research and education. Furthermore, we accept international students in Faculty of Veterinary Medicine, such as from Malaysia.
The reason I chose UGM is because it's well recognized and known for its academic excellence. My life in UGM, I would say, is tiring, but it's fun because I get to experience new things in class and also outside of class, such as my practical labs. The activities that I enjoy doing would have to be IVSF from the International Veterinary Student Forum, where we carry out activities such as Outbound, which allows us to not only bond with our seniors but also our lecturers.
I'm currently doing my 5th year in veterinary medicine in UGM. Why I picked UGM was because it's recognized in most countries, especially in Malaysia, and it's also in a city which can be very convenient. And the activities that I find interesting in UGM is the fact that the students get to teach the juniors a certain lab For example, I'm currently an assistant in the lab called Veterinary Clinical Diagnostics. So what I learned from my seniors and from my lecturers, I actually carried on and teach to the juniors so that they also get the knowledge that I received from my seniors. So that's what I find interesting about UGM.
In order to pursue our dreams, we have done various community service activities. The Professor Suparwi Animal Hospital at Jalan Fauna No. 2 Karangmalang, Yogyakarta is one of them. The animal hospital is equipped with representative facilities and professional management. This animal hospital can become the reference of animal health care in Yogyakarta and its surroundings. Faculty of Veterinary Medicine UGM is committed to developing science and community service based on the veterinarian work principle Manusia Mriga Satwa Sewaka, which means we are to serve mankind through animal kingdom.
Okay. Ladies and gentlemen, thank you for those of you who keep staying with us. Now we will move to the second invited speaker who will deliver very interesting topic entitled Host Pathogen Interaction and a Rise of Neglected Tropical Disease Due to Global Change. This topic will be delivered by Professor Carlos Hermosilla. He is a professor for parasitology and parasitic disease of the Biomedical Research Center, Salzburg Justus Liebig University, Gießen, Germany. Please, the organizer.
Dear ladies and gentlemen, first of all, I would like to express my gratitude to the organizing committee of the first ICAVESS conference to be held in Yogyakarta, Indonesia. And I'm very, very delighted to have the chance to talk about host pathogen interactions and the rise of neglected tropical diseases due to global changes. Now, before I start my talk, I would like to draw your attention on the following very simple scheme. Seeing here, where on the right side you have the human being and obviously exposed on a regular day to a diversity of invasive pathogens, including the ones occurring only in humans, but also to a wide range of parasites, viruses, bacteria, and fungi that are mainly generated by different animals and sharing habitats with humans. Now, if you take a closer look here on these numbers, over 60% of human pathogens are zoonotic. And within this 60%, 70% of them originated from wildlife species. And in the tropics, particularly, we have the highest biodiversity. And this means that humans living in the tropics are more exposed than in Palearctic regions, for instance. And obviously, this needs to be taken into account if we want to understand how diseases are transmitted in terms of transmission, as we don't have only vector-borne diseases, but also the ones that might be transmitted by ingestion. Now, humans, as well as all mammal species, and including also our vertebrates, insects, ticks, are able to survive due to their immune system. And if we keep an eye close to the humans and mammalians, this immune system can be divided in two branches, namely the innate and the adaptive immune response. Now, fortunately, we know quite well about the adaptive immune response, but on the other hand, innate immune response has been neglected for almost a decade. And for many years, it was considered a primitive and rather irrelevant innate immune response, but this is the opposite. So in the next slide, I would like to acknowledge the person who discovered this system. And this is seen here, Ilya Ilyich Mechnikov. He was the Nobel Prize winner for this marvelous discovery, where he described for the first time professional phagocytes and also cells that are participating in this innate immune system. Now, in addition to the professional phagocytes, you see here endothelial cells covering all blood and lymphatic vessels, as well as epithelial cells that are present in the skin, in the intestinal gut, in all mucosal surfaces of body openings such as reproduction tract, eyes, ears, nose, and mouth. So pathogens, once breaching these barriers, these physical barriers, are immediately confronted by professional phagocytes. Now, our interest is mainly on one particular leukocyte population, and this will be shown in the next slide. Here you see the polymorpha nuclear neutrophils, which are abbreviated in my talk as PMN. And why I'm so interested in this particular group of cells? Because they are the most abundant leukocyte population in the blood, the first ones to be recruited to the site of infection, and the first one to fight immediately to all kind of invasive pathogens. And furthermore, to orchestrate the adaptive immune response onwards. And here below, you will see the main killing mechanisms displayed by these cells, by PMN. Here at the left side, phagocytic activities or phagocytosis, degranulation, leading to the release of proteins and antimicrobial peptides, and the so-called neutrophil extracellular traps, abbreviated as NETs, where they are able to extrude DNA into the extracellular space and trap parasites or other pathogens. Now in this scheme, you see a bacteria, rather tiny pathogens when compared to nematodes, trematodes, or protozoan. But to be honest, we showed already very early in 2006 that PMN were also able to act against large-sized protozoan, such as apicomplexan parasites. And this is shown in the next slide, where you see a bovine PMN attacking and grabbing for a sporozoite by forming laminopods toward this parasite. And if you continue this cultivation, you end up having this situation where the sporozoite is now, after 30 minutes, stuck into the cytosol of an activated PMN. And if you continue this for hours, you will end up having the sporocytes intracellularly located. And the most striking feature is indicated here by the arrows, that you clearly end up having an impact or a disruption of the surface of this intracellular sporocytes, indicating that PMN are able to degrade and kill once phagocytized parasites. So we were aware that this professional phagocytes were very efficient in fighting even large-size parasites. Here's another aspect that I need to address before I start showing data. As I said to you before, endothelial cells are also participating on the innate immune response. Imagine an endothelial cells become activated by a pathogen. It immediately responds. They are highly immunoreactive and able to express a different type of adhesion molecules in order to allow free circulating neutrophils to initial tether to the surface, and then to slow down and roll, and then come to an arrest and firm adhesion. And if you continue this after 5 minutes, some of this adhered PMN might migrate between endothelial cells and reach the areas of infection. And this can also be seen here in vivo, where you have a vessel And in this vessel you have free-flowing neutrophils as indicated here. Some of them are adhered to the endothelium and others are already extravasiated and found in close proximity to pathogens. And this shows you how complex this innate immune system is. Now, if you go into the gut, you will have the same situation. Here you have the epithelium, and this drawing was provided by my PhD student Juan Vélez in a manuscript that we have just submitted on Cryptosporidium. And you see again that PMN can be recruited to the site of infection, and this can also be evidenced in vivo in Eimeria bovis infected intestinal mucosa, where we showed in 2016 the presence of various neutrophils surrounding, in this case, gammon stages of Eimeria bovis. The same occurred also for other closely related parasites in the gut of caprine. So in vivo evidence proved that this is also occurring during disease. Now coming back to the last effector mechanism, neutrophil extracellular traps. In here you have a very simplistic approach where it shows you the vital and suicidal netosis process. You have on the surface of PMN various, as you see, receptors and molecules, and here on the left side you see that, for instance, antibodies or PMA or cholesterol crystal can, for instance, activate NADPH oxidase, which leads to ROS production, and this ROS production activates PET4, which leads to the chromatin decondensation, and the decondensation will allow to be released into the cytosol and mixed with granule contents such as myeloperoxidase, neutrophil elastase, LF37, various histones, and then after the disruption of the PMN membrane, these structures are released into the environment. But we have also vital netosis, which is a bit different, where mainly the DNA originates from mitochondrium. Also here seems that ROS have a pivotal role, but independent of ROS, irrespective of ROS, you have here a key molecule, PET4 mediated citronylation of histones, allowing the decondensation and the release of the structures, the sticky structures. Now, the PMN still are able to perform phagocytic activities and chemotaxis. So this needs also to be taken into account. This was published in Nature Medicine a few years ago. Now, I would start showing you the pictures of the first publication of NETs, which was published by Brigman and colleagues in Science. And this was a landmark study because it showed that PMN, as I said, by doing NETs, they are able to kill pathogens far beyond the lifespan. And the properties of NETs are one illustrated here on the right side. They are membrane protrusions composed of DNA fibers, very thin ones, globular domains, granule proteins and peptides are decorating them, as seen here in E. Here below you see this granule contents, and they are very filigrane and able to bound to pathogens. Now, as I said, this was a very, very interesting publication because it broke a paradigm that cells could kill pathogens far beyond their lifespan. Soluble factors were also described in these publications that were able to induce NETs, but more importantly, they showed that some bacteria, seen here, were able to induce NET formation and leading to the entrapment of this bacteria, but not only to the entrapment, but also the killing of this bacteria. As you know, bacteria are very tiny in comparison to a protozoan or a metazoan parasite. So when we read this publication, we thought if our PMN that we were working on could also act against parasites, and we were lucky that we had at that time the parasite Eimeria bovis in our hands, and the first experiment showed very clear that also parasites were able to induce netosis. And actually, we are very pleased to say that and proud that we were the third group in the world showing that parasites were able to induce NETs. The first publication was by Baker, showing that Plasmodium falciparum malaria was able to induce in humans netosis. The second one was by Gamarange, a Brazilian group, on Leishmania amazonensis, and we, with this bovine Emeria bovis parasite of cattle, and here you see the streets of fibers entrapping different sporozoites. Now, if you take a closer look to this area, you end up having again these filigrane structures, sticky structures covering the sporozoites, and as I said, they are a combination of DNA and antimicrobial peptides or proteins and histones. We then next tried to quantify these reactions, and as seen here on the right side, we showed very clear that viable or vital sporozoites induce a very strong reaction when compared to the negative control, which are seen here at the right side. And the most striking features are here in the middle, where it shows that heat-inactivated sporozoites and homogenated sporozoites, that means antigen-soluble antigens, also induce strong reactions on a significant level when compared to the negative control. And our positive control in this case was PMA, it's the one used by Brinkmann et al. And here at the left side you see once again the net structures entrapping two sporozoites indicated by the white arrow. We then wondered whether nets might kill the parasite, and to our surprise, and in contrast to the findings of bacteria, we couldn't see dead parasites. So we wondered what could be the action of these nets against these intracellular parasites. And for this we did a very simple experiment seen here. We confronted our endothelial monolayer where the parasite replicates and exposed them to vital sporozoites of Emeria bovis, and they reach approximately 45% of infection rate, the ones that we are usually seeing. Repeated the same experiment, but now in the presence of PMN and the release of nets, there was a significant reduction of parasite invasion. Now everything is reversible as soon as you start adding DNAs into your medium and allowing the sporozoites to get free and become active and infect again cells, and almost reaching the same levels as the ones alone. So it showed for the first time that nets not only entrap, but they also inhibit the invasion process and probably the dissemination of intracellular parasites. And this was a nice finding. We also have published a few works on metazoan parasite, and as an example you have here Dirophilaria immitis, a very important zoonotic parasite occurring mainly in dogs, where we observe societal and aggregated nets. As you see, there are many phenotypes. And in here at the right side you have a microfilaria entrapped in a very compact structure, and these are called aggregated nets. Below you have also the vital spread of nets, where we expose PMN, in this case from bovine, to tachyzoids of Besnoita besnoidi, and adding again some features and showing vital netosis. I brought here this movie to show you the role of it's not working. Well, actually it should have been a movie where the larva is entrapped, but it's not working, and was able to be trapped. Unfortunately, it's not running. Now, when you do a closer look on this larva, you end up having other phenotypes such as suicidal class-like structures, and here on the right side you see the overlay showing DNA decorated with cytox orange and different histone types, and thereby confirming again that these are really nets. And if you continue this incubation process, you end up having this beautiful aggregated net formation, ending in a completely entrapment of a larvae, and this larvae can't escape. This is something evident and clear. Now, to show you a bit more, unfortunately I can't bring the movies, it's a pity because I had movies. There are, as again, this suicidal netosis also in Dirophilaria immitis exposed parasites. Now, to show you that nets are this effector mechanism is well conserved evolutionary, we isolated PMN from dolphins, seals, and other species, and it shows you how conserved and how ancient this system is. And here, PMN of dolphins were exposed to tachyzoids of Toxoplasma, and again, resulting in different phenotypes, such as aggregated, diffuse, and spread nets. And I have to say that the strongest reactions and the fastest ones were seen for dolphins. After 10 minutes, we had already very strong reactions concerning nets. Here's again another movie that I can show you where vital netosis is shown. Coming to another aspect, now netosis might occur also in vessels, and this is shown here, and this was published in 2013. So we wondered whether we could also repeat experiments, but now infecting endothelium, primary endothelial cells, and then look for net formation under flow conditions. And this was done, and as you see here in the panel above, you see at the left side scanning electron microscopy picture endothelial cells infected with the tachyzoids, here the intracellular stages, and the PMN releasing structures, nets, and here on the right side you see again nets occurring under flow conditions. But the most important thing is that we could identify also molecules responsible for the induction of netosis, such as CXL8 and P-selectin. And here below we quantified and showed that in case of besnoid infected endothelium, there was a significant increase in PMN adhesion when compared to non-infected cells. And here on the right side again our positive control, which was endothelial cells stimulated with TNF-alpha. So they really induced activation of endothelium, and this leads to the protrusion of nets under flow conditions. This is just to sum up this first part of my talk and hopefully showing you how important it is to take a bit more serious the innate immune response and discover the beauty of it. And in 2004, as I said, I showed you the first publication by Brinkman et al. Our work came here, as I said, the third one. And by 2017, we had almost 400 publications. It shows you the importance and the recognition on how nets are not only dealing with parasite, but they're also influencing pro-inflammation. They are involved in tumor progression and many other diseases, even reproduction. Now coming to the second part of my talk, climate change. And as you know, I'm a fan of facts. I hate fake news, and this is something that we can't deny. The land and ocean temperature percentiles in July 2020, and you can do it you can go into the PubMed and check all the regular meteorological countings, and you have a significant increase in temperatures, particularly in the tropics, seeing here, and in Southeast Asia. And here's Indonesia. And you see here in these areas we have a record of the warmest years. And I'm pretty sure that I don't have to convince that this will have an impact on many factors, as this is one single world. But the hope is seen here. The youth have recognized the importance to change this dramatic impact. We need to do something, otherwise we will end up having very much destroyed our planet. Now, these climate changes have impact obviously on different scenarios. I'm a parasitologist, so I'm focusing more on the biological aspects, and here you will have an impact on certain organisms that are clearly adapted to higher temperatures. And as soon as you start invading new areas, for instance, the tropical areas are increasing to the north and to the south due to climate changes, you will have also the introduction of these vectors into new environments, and therefore transmitting diseases into animals and humans. And this is just a simple scheme showing that climate changes will also have an impact on many other aspects as seen here. Now to be more precise, I just brought here a very nice GIS picture where again you can add the temperatures. In red you have the higher the higher transmission rates, where you can extrapolate GIS temperature and calculate risk factors. And as you see here in this map, the highest risks are where you have more increase in temperature when compared to other zones. And this is due, as I said, to the spread of mosquitoes into these areas. And to bring it to a global scheme, I brought here this nice global atlas of lymphatic filariasis by a period of time, published in Parasite and Vectors, as you see here. And the fact is that you can, in red, you see how fast it has spread into areas non-endemic and to previously non-endemic areas. Again, due to the fact that we are allowing mosquitoes to grow better and develop better in these areas, but and also the migration of people has been taken into account. And here is Indonesia, your beautiful country, and as seen here in Asia, you have now increases in other parts of the world, including areas that were not considered including countries such as South Korea, which is not in the tropics, and the same is in Africa and in Asia. Now all this has to be linked to the neglected tropical diseases. As I said in my introduction, in the tropics you have the highest biodiversity on species, and obviously also with their pathogens. And in the tropics you will obviously have more neglected diseases occurring and affecting, unfortunately, the poorest of the poorest. And in the tropics, over 1 billion humans are affected by more than one entity seen in this global map. So I think we need to address this aspect in our future surveillance studies. Now coming to the pathogens, as I said in my introductions, we have virus, bacteria, fungi, and a lot of parasites, including ones that are transmitted by mosquitoes, such as lymphatic filariasis, onchocerciasis, but also gastropod-borne parasites, such as schistosomiasis, fasciolosis, and others, and soil-transmitted helminths. And here also some others that are transmitted by tabanids or other insects. But for me, it's more important to show you this next slide, because Liu et al., and this was published in Lancet, shows the global trends in burden of childhood death from 2000 to 2010. And here on the right side you have the rotavirus, the most important pathogens causing death in children, very young children under the age of 5. Rotavirus on the first place, Cryptosporidium parvum and Giardia intestinalis, 2 parasites. I mark them in red to emphasize on their zoonotic potential. Now, and this is constantly seen, but we are not taking care of it. We are ignoring. And if you take into account only Cryptosporidium parvum infected children and Giardia intestinalis humans, altogether, they are larger in numbers than the whole neglected tropical diseases. So this is something to worry about and to be considered. And the other fact is seen here. You don't see very much, but in the next slide you will understand why. We call this planet Earth, but how inappropriate it is shown here. You as Indonesian people are aware that we are surrounded by an ocean. And the fact is that over 70% of the Earth's surface corresponds to water surfaces. And more than 96% of all water sources on Earth are contained within oceans. And what do we know about the ocean? Practically nothing. Approximately 19% of the species are known so far, and so there is a lot of work to do in terms of understanding. But the most important thing is seen here. Anthropogenic pressures on ocean plays a key role on climate as well as global changes. The global changes are directly linked to currents like El Nino or the Gulf current and others. And the other aspect is seen here. We have so far, and you are pretty aware of the One Health concept, but this is focusing mainly on terrestrial animals. And what about the marine ecosystem. And I'm very pleased to have the chance to collaborate with the Nobel Prize winner Jonathan Pfaff, who actually introduced to the world the global health instead of using the word One Health and Planet Health, because he thinks that this is a blue planet and that we need to take care of the marine environment. And I'm here seen in close proximity to him, and not because I want to get all his thoughts, but just to highlight that he actually acknowledged our work in Gießen on marine mammals by giving a presentation at our university and wearing a tie, and if you take a closer look, with whales. And because we have done quite a lot of work on this. Here, this is our working group on marine mammals. And here I would like to acknowledge my wife, Anja Taubert, Liliana Silva, who is actually also here present at this meeting, Juan Vélez and David Ebma, which are all working now in this. And we are focusing on different marine mammals, not fish, and to start working by a non-invasive approach in order to make sure that we don't disturb these animals. And briefly, anthropogenic neozoan parasites are already occurring. And you see here Toxoplasma gondii and Cryptosporidium parvum, again, a very important parasite. And both oocysts are highly resistant. Apicomplex and oocyst are very resistant in the environment. And Cryptosporidium is considered a waterborne disease. And this situation where you have calves in close proximity to the shore is not a seldom issue, and the same is true for felids. Now, this is our philosophy. We work on non-invasive or minimal invasive methods by investigating these mammals, and we started this work by sampling and diving with dolphins in the Red Sea. This was published in 2014, and here Sonja Kleinert, my former postdoc, who is also attending this conference, successfully collected fecal samples from free-ranging dolphins, and we discovered that they had a variety of parasites, including Giardia infections in this scenario. So these pathogens are occurring in the environment. And here is just to show you another example of how a non-invasive method can be used. Here my doctorand is combing some parasites from sea lions, and again you see the close proximity of humans with wildlife. And at the end, I would just to show you that this can also be done in whales. We collected fecal samples from blue whales, fin whales, sei whales, all alive, and sperm whales. And when analyzing the fecal composition, we found that, for instance, Entamebia, Giardia, and Neobalantidium were present three zoonotic parasites. And believe me, 180-ton blue whale don't defecate just a few grams, they defecate tons. That means there are 100 billions of Giardia and other parasites in the ocean, and we need to address this if we have to consider the future. The same occur when we sampled endoparasites and we looked for endoparasites occurring in sperm whales in the Mediterranean Sea. Again, here in red are marked the waterborne parasites, Giardiosis and Balantidiosis, but also nematodes such as Anisakis and Diphyllobotride, which are also human relevant, were detected in these animals. And this is just a summary where it shows you that we were actually the first one describing in whales, in large whales, the presence of important and neglected parasites. And I said Giardia and Cryptosporidium are very, very neglected. And this needs to be changed in the future. And at the end, I would like to bring the last and most important picture here of my talk, namely our group. I would like to acknowledge my wife, Anja Taubert, and all the postdocs and doctorands that I have at the moment working on nets, but also on wildlife, as I said. But most important is also to acknowledge the foundations who support this research for many years, DFG, the German Research Foundation. And on behalf of myself, I thank you for your attention and once again for your kind invitation. Thank you. Good morning. Can you hear me?
Yes, we can hear you, Professor Carlos. Thank you for joining with us. Also,
It's a pleasure for me. It's a pleasure. It's a pleasure.
We have many questions.
Yes.
For you. So
Yes.
we will
Feel
show
free
the,
to ask them.
Question, and I hope that you can directly answer the question.
Yes, sure. So, which is the first? Thank you.
The first question from Professor Wayan Tunas Artama. Okay.
Yeah, it's a very, very, very important question. Actually, we are quite convinced that that there are some ways to understand how, for instance, macrophages and monocytes are able to participate also in this type of reactions. But on the other hand, as you said, the question was related to parasites that they can survive within the phagosome. But there are interesting new aspects that, for instance, certain activated cells, such as macrophage, can recruit other cells to get some help in order to work together. I didn't show all the interactions between the different cell types, because you don't have only PMN circulating the system or in an organ. You have monocytes, you have also the macrophages coming in, and the same is occurring if some pathogens might overcome the first barrier, then comes the second line of cells in trying to avoid and kill them. But to be honest, we haven't so far addressed macrophages in our studies. We are working also monocytes, and in there we will continue on something called the that allows you to interact with other cells. But in terms of macrophages, we don't have done so much in the past. Is there another question?
The second question. There are.
I can see it. At the moment, in Germany, we have different organizations and groups dealing and working with wildlife animals. And one is the Leibniz Institute. It's a German central institution which covers a lot of research on wildlife. But we have also this, again, Leibniz also dealing with large and livestock animals in the northern part of Germany. So there are central governmental supported institutions, but we have also local in each federal state a vast amount of groups and courts working and investigating on animals, but not only in physiology, but also on diseases. But in terms of marine mammals, we have only few institutions and all linked to faculties. For instance, in the north of Germany, there's a center dealing with the northern Wadden Sea. It belongs to the Hanover University, to the veterinary faculty of Hanover. In our case, we don't have any court for this type of investigations.
Thank you for Professor Carlos. The next question, please.
Oh, I'm very, very thankful for this question. If you go into the PubMed, you will find now, I think, at least 30 publications showing very clear that nets are occurring and actually being involved in the severity of SARS-CoV-2. And initially, we also intended to start our research also with some virologists here. But as you can imagine, at that time when the pandemic started in Germany, it was very difficult to get samples, but there are evidence. So I'm very pleased for this question. Actually, I have to admit that due to the fact that the pathologists started to look into the diseased persons, they identified clearly the netosis in the lung. And what you have here is just an overreaction of endothelium, of epithelium, releasing cytokines, and this leads to afterwards to the release of netosis. But they clearly showed and linked netosis to coagulopathies seen in the lung, to the severe damage. And the same is occurring in organs, in other organs from COVID patients. For instance, there are in Brazil some patients that showed epididymitis, orchitis. And again, when they looked into the pathology of this reproduction tract, they found a vast amount of netosis. So yes, it's occurring.
Thank you, Prof. One more question, I think.
Yes, please.
Please.
I think this is a very, very important question. The first thing is that we have to, first of all, to identify really the clearly ones affecting the world. And as I said to you before, many pathogens are there, and they are ignored. And this is done often because the first world is dealing in leading the research. And I'm very honest on what I'm saying now, and it will be criticized by many people, but I have to say it. For instance, there are many groups in the world working with leishmania, which is important, obviously, I understand, and obviously also in Europe and in America. So they have a sort of created a very important consortium leading or bringing a lot of people of the first world dealing with this disease. But it's not the most important one if you go into the tropics. And that's what I just explained very briefly. If you take just Giardia, crypto, and rotavirus together, they overcome the numbers of all neglected diseases. And this can't be continued. And the same is if you go to Africa, there you will see other diseases. Soil-borne transmitted disease, Ascaris, Trichuris. Who cares about them? Nobody, but everybody's talking about schistosome, leishmania, and whatever you mention. And I think we have to consider first and clearly identify the ones with the real impact on health, and particularly the affecting the youth. Because if you hit the youth, you don't have a next generation. And I'm very pleased to announce that we are working now in Cameroon on Cryptosporidium parvum and other diseases that are occurring in childhood. And I think this is something that needs to be done. I'm working here on neglected diseases. I'm part of a consortium, a very big one in Germany, where we address neglected diseases, and we are actually just a few doing or dealing with diseases that are really affecting the vast amount of people. We have also colleagues working on diseases that maybe they might bring together in the world 400,000 per year, the incidence, some bacteria, for instance. I don't want to name the bacteria, but this is what I'm saying. You have to, in my opinion, to identify very rapidly the ones really affecting the world. And it's very easy to combat them. For instance, Cryptosporidium, if you take care of water supply, hygiene standards, education, it's easier than to start looking for drug development and whatever. It might take 100 years to get a proper drug against this parasite. Instead of saying, okay, we have to address, as you said in your video, bring in the knowledge and educate the people. Don't drink just dirty water. This type of things. Malaria can be easily combated by bringing mosquito nets. And this is something that needs to be addressed. But as I said, bringing in also the people that are affected. So what I want to see is more active participation of people living in the tropics with their problems. And in case of Cameroon, we have a colleague from the University of Buea, and he is the leader there. So this is also important to have both sides, particularly the ones working in the tropics, to bring this knowledge and spread this into the community.
Thank you very much for the
You're welcome.
explanation.
You're welcome.
Due to limited time, we will move to the next presenter. Thank you very much, Prof. Carlos.
Thank you
for joining with us.
It was a pleasure for me. Thank you very much.
And great to Anja.
Thank you. Ladies and gentlemen, the next presentation will be delivered by Assistant Professor Dr. Chenpop Sawangmake. He is the person in charge of the Veterinary Stem Cell and Bioengineering Innovation Center, Chulalongkorn University, focusing on stem cell biology and translation, bioengineering, tissue engineering, and regenerative medicine for human and veterinary practices. Today, Dr. Chenpop will present about stem cell in vet, from science to future hold to make a better world. As we know, this topic is very futuristic, and we will learn together from him about the update of stem cell research in veterinary practices. Please kindly send your question in the chat box, and please pay attention to the question format that will be informed by the organizer. The organizer, please show the video.
Dear all participants, I am Chenpop Sawangmake from the Veterinary Stem Cells and Bioengineering Innovation Center, Faculty of Veterinary Science, Chulalongkorn City, Bangkok, Thailand. So first of all, I would like to give my sincere thanks to the organizing committee for inviting me to join the first International Conference of Advanced Veterinary Science and Technologies for Sustainable Development 2021. So the topics of the talk today is about the stem cells in veterinary science to make a better world. So first of all, we'd like to start from the impression that I've got during the research career. So we talk about the Nobel laureates. There's a lot of people that got the Nobel Prize. However, we will look at the people in field of stem cells. We have several people that achieve a big impact on the stem cells research now today. Starting from 2007, three of the laureates, Professor Mario Capecci, Professor Martin Evans, and Professor Oliver Smith is shining and sharing the prize in terms of Nobel Prize in Physiology and Medicines in 2007 for the discovery of the technique to produce transgenic animals, which is a big challenge and a big impact for scientific research right now. At that time, they are invented a technique that use the embryonic stem cells that is the cells that got from the blastocyst stage of the embryo, and they take the inner cell mass, grow them, and then they produce the construct of the gene that they want, and then transfix or insert the gene into the cells. Finally, they get the cells that contain the target genes that they want. Then they grow the cells and use the selection method to select the pure cells and make the whole clones of the cells. By this means, they got the embryonic stem cells that contains the genetic materials that they want to study. Further, they transplant these cells into the embryos in the state of blastocyst, and then they put this embryo that contains the egg cells with the target genes into the surrogate animals to be the host to make the offspring. By this means, they can produce the transgenic animal that contains the genetic material or the genotype, then transfer the phenotype that they want. Right now, this technique is very, very popular, so because we can produce transgenic animals that contain the gene that we want to study, which is a big challenge for study right now. And then around 5 years later, two Nobel laureates, Professor Sir John B. Gurdon and Professor Shinya Yamanaga, sharing the prize in Nobel Prize in Physiology or Medicines in 2012. Professor John B. Gurdon is the first one who invented the technique for cloning the animals. Professor Gurdon used the eggs of the frog and then take out the nucleus and put the new nucleus from the cells of the tadpole, and then he grew the cells, and then the cells become to be tadpole and becoming the frog. This technique is a platform for cloning for today. This is the starting of the cloning of many of species, including the dolly, the sheep, and also many of species. Then Professor Yamanagar invented a technique that can reverse the somatic cells or adult cells by reprogramming the cells into the states of the cells that very alike the embryonic stem cells. By this means, we can produce the induced pluripotent stem cells or iPS cells, which contains the property like embryonic stem cells. And these cells theoretically can be differentiated or induced. This is the big challenge for the new study of stem cells for today, because no need to destroy any embryos or living organisms. We just harvest the adult tissue or adult cells and do some of technique reprogramming and then we can get the cells that contain the potency property for making other cell type. So after I got the PhD in veterinary bioscience from the Faculty of Veterinary Science, Chulalongkorn University, I got more training from the research unit of mineralized tissue and the Center of Excellence in Regenerative Dentistry from the Faculty of Dentistry, Chulalongkorn University. And then finished the course of Fulbright Research Visiting Scholar from the Department of Bioengineering, University of Washington, Seattle, under the supervision of Professor Shishichi Ashley and Professor May Spear. So I came back to the country and established their innovation center, so-called Veterinary Stem Cells and Bioengineering Innovation Center. Right now we have the members around 15 to 20 members working on stem cell research and innovation. By the close support from the seniors, researchers and lecturers here, Professor Simthonjip Shok Anand and the Dean and the Vice Dean, and two professors from the Center of Excellence in Regenerative Dentistry, and from many of colleagues from the medical school and engineering school, with close support with my senior mentor, Professor Ashley from University of Washington, Professor Hiroshi Egusa from Turku University, and the colleagues from the Canal City, Professor Santiago Palata and Professor Nadine in the oral unit of the Canal City. So we are working to establish the WISPIC, or the Veterinary Stem Cells and Bioengineering Innovation Center, that aims for invented the innovations and the platform to treat the disease using stem cell based technology. So right now, our lab, our research center, focusing on three main aspects of research. First is about the diabetes treatment. So we are trying to use the bioengineering platform to produce insulin producing cells, and when we try to permanently treat the diabetes type 1, start from animals, and we aim for adapt this technique to be used in humans also. Second one is the themes of the bone tissue engineering. We aim to use technique to treat the bone defects in terms of the craniofacial defects and the skeleton defects. And the third one is about the eyes defects. We are working on the project based on this corneal film and stomach scaffold. So starting from the work that we are working on, so we are trying to use the technique of the genetic engineering and bioengineering to produce a cell platform for us. And we try to use many sources of stem cells, like a cell from dental tissue and a cell from bone marrow and other type of cells. And then we try to do the banking. Right now we have quite a big lot of cells banking, and then we transfer this knowledge to produce the platform to treat diabetes in terms of producing the cells that can produce insulin, and to develop the delivery system to transplant the cells into the host. And we are working on the bone project and also the eye project. So we're focusing on the diabetes team. So we have Dr. Wachuriwan and Dr. Suryo as the leader of the team. And right now the member we have Dr. Hong Thuan Thorn from Vietnam, Dr. Dreta from Indonesia, and Dr. Lê Dong Quynh from Vietnam. And also we have newcomer Dr. Irmar Pradita from University of Gajah Mada Indonesia, starting as a new member, and we have the research assistant, Ms. Korenit Patanachai, working on the diabetes project. So we are trying to propose a new way to treat diabetes permanently. We are focusing firstly with the diabetes type 1 from animals. We're focusing on the canine model first. So we in 2020 we proposed the ideas on the productions of insulin producing cells with a transplantation platform to be applied in the veterinary practice. So we proposed the idea to produce the insulin producing cells or the beta cells in vitro by mimicking the development pathway of the organogenesis or pancreatogenesis, starting from the cells or the stem cells, and then we drive to mesoderm, to definitive endoderms, and then forward to pancreatic endoderm, pancreatic endocrine, and then we're gonna get the mature insulin producing cells or mature beta cells. Sometimes we call the cells as islet-like cells. By this means, we can produce the insulin producing cells in vitro, and we are trying to develop the delivery systems. We proposed a way to use the encapsulation platform to hold the cells, preserve the cells, to make the cells or the colonies stable, not stolen, not leak, and can store within the beads of encapsulation. This should be a very good biocompatible property, which is very challenge for us to find the materials or the combination of materials that suit the biocompatibility properties. And we hope that this encapsulation method can prevent the cells from the invade of the immune cells, and it should provide the nutrients and oxygen supply to the colony insides. So when we achieve this goal, we will have the tools for treat diabetes type 1 in animals, especially in canine diabetes. We first proposed the idea in 2014 in the BBRC. We proposed the first time that we can produce the insulin producing cells from adult stem cells. At the time, we used human data pouch stem cell as a role models with the collaboration work with the Faculty of Dentistry Chulalongkorn City. So we use the multi-step protocol to induce the cells, like the pouch stem cell or the periodontal ligament stem cell, which is the somatic stem cell, into the insulin producing cells. So at day 10 of induction, so we can get the round colonies of the cells, which is behave like the beta cells in terms of genetic and their phenotype. So we see the set of the pancreatic genes markers application comparing with the control in both cell type. And then when we stain for insulin protein within the cells, we see the expression of protein of insulin within the cells. And then when we test the functions, we have the good functional property of the insulin producing cell that we produce in vitro. So this is the first time that we are able to produce the insulin producing cells in vitro from the adult stem cells. And then in 2020, we proposed the second papers in BBRC mentioning the integrative protocols to improve the efficiencies of the cells differentiation. By these papers, we propose many of the way to enhance the cell differentiation, starting from the pure reprogramming of the cells, or we combine the reprogramming with the microenvironmental manipulation of the cells, with the variations of some of the signaling manipulation like notch signaling. So by this means, we have a variety of results which give us a very good cell properties and colony size that are suitable for transplantation and for their ability for working here. And then we have the good expression of the pancreatic markers in terms of the progenitor markers, the mature beta cells marker, and the relative markers. So then in another project in 2020 by Dr. Suryo, we invented the way to preserve the cells by encapsulation technique. We used a mixed material from the arginate and the puronic F127 based encapsulation. So we show that this method can support the cell viability and also the functionality of the iPSC cells that we get it from hematopoietic stem cells. So Dr. Suryo showed that the encapsulation method can preserve the viability of the hematopoietic stem cells in long-term storage or culture, as shown in their lab date staining and the alarmable staining. And then when Dr. Suryo did the encapsulation of the insulin-producing cells, so the cells show good morphology within the capsule, and then we check the live dead staining. So we have more than 80% of the cells that are alive. And then we check the expression of the insulin protein and proinsulin protein, and we found a very positive result since day 14. And then we check the cell properties or the functional property, we get a very good result comparing with non-encapsulation method here. So we have the quite exact the same functional properties of the cells comparing to the non-encapsulation cells. So by this means we can conclude that to produce the insulin-producing cells from the adult stem cells is feasible, and we can preserve the cell property by using the encapsulation method. Right now we are submitting the manuscript about the generation of insulin-producing cell from canine cell resource. So we start from the bone marrow cells and adipose cells. Right now it's under the revisions from the journal scientific reports. And then it's about the bone tissue engineering work. So this team led by Dr. Suratan Tawisai, and I remember we have Dr. Menadia Pervaningrang from the University of Gajah Mada, and Dr. Nebilar Jamilar, Dr. Steven Prabantoro from Indonesia, and we have the scientist support, Ms. Piyuwan Charan-Lertgun. By these topics of the bone tissue engineering, we are trying to propose and to establish the MSc-based bone tissue engineering for veterinary practice. So these review articles by Dr. Surat proposed that the stem cell-based bone tissue engineering required sets of bone markers to check. So we found that most of the reliable markers include the RANKS2, the osteocalcins, the collagen 1A1, the osterix, osteopoietin, and ALP. And then we found that the three stem cells, which are mesenchymal stem cells, that could be able to use in bone tissue engineering are the cells from bone marrow, the cell from adipose tissue, and the cell from the hematopoietic stem cell. So they have a different advantages and disadvantages. But however, at that time we need quite a lot of study to qualify their characterization. And we proposed a way to enhance some property regarding the cell proliferation and the potency of the cells. We're starting from the bone marrow cells. We found that with the treating of the simvastatin, which is the drug that used in humans, we can enhance the perfusion property of the cells at day 7 and also day 5. So this is because of the reducing of apoptosis of the dosage of simvastatin, that not all the dosage of we found that to use simvastatin as the treatment of the cells, we need to vary the dose quite a lot. That is still a challenge for us to apply this in the clinical practice. So recently, we proposed the idea in the papers that got published in the scientific report. So we use the technique of system biology to clarify behaviors of the cells from kinetic resource. So we focus on the osteogenic differentiation behavior of the cells and covering the cells from two sources, from bone marrow and from dental pulps. So we compared their properties and we found that they behave quite different. The cells show different sameness markers, different percentage of the surface marker, and also different properties of the CFU forming. And then when we qualify the properties in terms of the adipogenicity, chondrogenicity, and autogenicity, they show some difference in their properties. When we look deeply at the property regarding the bone induction, we found that the bone marrow cells show less efficiency compared to the deposit stem cell in terms of the bone induction property. So we found that the bone marker, when we induce the cells, the deposit stem cell into the bone cells, they show quite high levels of the bone marker like RANKS2, also calcins, osteopoietin at day 7 and day 14. And when we do checking this mineralization of the cells, we found that the deposit stem cells show a better mineralized tissues regarding the induction protocol. And then we use the proteomic analysis to clarify the behaviors. So we see the distinct pattern of protein expression between two cells, marrow cells and the deposit stem cells, between the 7 and the 14. So when we do clustering, we found that both of the cells behave differently. And interestingly, they are only proteins that co-expressed by the two cells, and there are a lot of proteins that expressed or downregulate in different way from both cells during the induction period. And when we check it with the heat map, we found that when we categorize the protein set into the signaling based on the David and Rectome platform, we see that both cells behave differently. So the first two columns is the marrow cells, and the last two columns is the deposit stem cells at the 7 and the 14. So the red color refers to the upregulation, and the blue refers to the downregulation. So we found a very, very different pattern of the protein expression from the two cells. So by this means, we conclude that these two cells requires very different pattern of the signalings for enhancing the bone induction. So for the bone marrow cells, it requires or it depends on quite many of the signalings. Signalings can work via SMAT 1, 5, and 8. With beta can work through and also. And the actuation of the Wnt signaling can enhance the bone induction in these cells. Why not signaling stimulation block the actions of the cells. But we look at the deposit cells, this behave differently. Only the BMP signaling can significantly enhance the induction of the cell toward the bone lineage. However, when we stimulate the TF-beta or the Wnt and also the Notch, it inhibits the bone induction of the cells. So this is quite interesting information that we got from the system biology analysis. So we found that these two cells behave differently. So the way that we work with them to apply these cells into the bone tissue injury. So in terms of the eyes defects, so we are working with Dr. Chudras Tosa Gun to making the. So we working with the team from the engineering school to use protein source to make the corneal films and the stromal scaffold, and then we are able to isolate the cells from the epitheliums of the corneas and from the stromal tissue of the cornea to produce the 3D materials that can use to transplant and replace the real corneas of the canine defects. And the last project that we are working on right now is about the food production. So we work closely with the private sectors, so-called the Thai Food Group. The Thai Food Group is a company that working on the food productions. So we are working to produce clean meat project, which is a lab grown. Has been started for a year, and we are able to produce the first prototypes of the pork-size lab meat in our lab, and then we are working right now to expand the knowledge and getting more collaborator to produce the cells that very look really alike the real pork meat to use as a food for people in the future. So right now we are working hardly to achieve our goal in terms of the productions of the stem cell-based platform to treat many of diseases in terms of veterinary science. And then we are working closely with our friends from the human health sector, trying to transfer and apply this knowledge into the human health or applications. And by the same time, we are trying to get involved in many projects that we think the stem cells will be the good tools for solving the problems. So in final, I would like to express my thanks again for the organizing committee for giving me to sharing our work, and then we hope that in the future we can produce a platform that have a very high impact for veterinary practice and also human practice. And we are welcoming every collaborations or every friends for joining us to make this dream come true. So thank you very much for your attention.
Thank you very much, Professor Dr. Chenpop Sawangmake. Thank you for joining this session. And also, I would like to thank you because you allowing our lecturers study in your laboratory.
With my pleasure, Professor.
There are many questions, I think, and now I would like you to answer that question. So organizer, please review the question. The first question.
Okay, it's about the advice and deterrence for studying the laboratory about stem cells. So actually, I think it's quite interesting that the way to start the lab is not easy in the past, but in right now I think it's more easy. We have a lot of experts working on stem cell research, and we have many of collaborators. I think we may start from joining as a like a collaborator or the colleagues, and then we can share the technique, anything that we can. And I think after COVID-19, maybe the exchange of the researchers for training might be the good way to building people for working on stem cell research and expand more collaboration.
Dr. Chen, Bob, how about the budget?
For
where the budget come from?
We have the budget from government sector plus private sector.
Also from private sector.
Both of the sectors, Professor.
I see. Okay, then the next question.
This is about the. I think it's my target on the specific cells or specific site therapy. And this one is the things that we are interested in. Right now, the technical is quite difficult, but in theoretically, I think we can do some platform to directly transfer the medicines or the reagents to the target cells or organs. I think it's still possible in the future, but need quite a lot of fundamental study.
Thank you. The next question, please.
It's about the bone graft from HA. Yeah, for sure. The hydroxyapatite platelet cells might be the good way to build up new bone. Eventually, we need to confirm in clinical study because in some materials with some cell properties, we cannot get the good results in the lab animal models. So we need a lot of little bit modification of the materials. Right now, we are focusing more on the another part of material like nanoparticles or some titanium particles mixed with the cells. That may be the good choice for regenerated bone and maybe the cranial tissue defects.
Okay, the last question, please. Still will have one question.
Yes, Professor. For the reproductive problem in cattle, especially for the hormone, right? I think for the stem cell research, we work closely with the field of the reproduction. We use a lot of the reproductive technology like in vitro fertilization or embryo transfer, and I think in the future we may be able to. I'm not sure about to produce the oocyte or sperm in vitro and do some purely, 100% in vitro fertilization without any animals, and we can do the transplant of this in vitro form of the fertilized oocyte in the surrogate animals. It might be possible. It might be possible, in my opinion.
Okay. I think this is the last question. So we will finish this session. Thank you for Dr. Chenpo. Thank you for coming.
Thank you very much, Professor. You're welcome.
Once again, thank you for Professor Rachel, Professor Carlos, and Assistant Professor Dr. Chenpo Sawangmake for joining the session of this plenary session. It was a pleasure to have you with us. Ladies and gentlemen, this is the end of the plenary session 1. For brief conclusion, due to global change, there are impact of infectious disease and also a rise of neglected disease that we have to aware. So innovation, including in-depth study and exploration in the veterinary field, as well as the other related field, is needed, including development of future therapeutic agents such as stem cell based on the synergy on science and the concern of sustainability. Once again, thank you for the wonderful presentation from the invited speaker. Thank you all for attending. We hope you have learned and enjoyed this session and have a great time in your parallel session. So I will give the session back to Karisma as the master of ceremony. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you so much, Professor Teguh. This is indeed an interesting discussion. We'd like to also thank you, Professor Carlos, joining us from Germany, and also Dr. Chenpo, joining us from Bangkok, Thailand. Thank you. And we are glad to announce that there are more than 200 participants joining us today. So to memorize our moment, we'd like to invite our invited speakers and also our distinguished guests to turn on your camera, because soon after this, we are going to have a virtual photo session. There will be more than 1 pages. Therefore, we'd like to ask your patience so our team can capture our moment accordingly. I will start on my count. For all the team here, are you ready? And also all the participants, for the invited speakers. Okay, so we'd like to invite all the deans from AFKHI or Asosiasi Fakultas Kedokteran Hewan Indonesia. We also like to invite our speakers for today, Professor Carlos Hermosilla, and then Assistant Professor Chenpo, Professor Omar, Professor Dato Dr. Muhammad Khair bin Bejo, and Dr. Pan Tong Yu to join us and turning the camera on. Okay, so are you ready? On my count, in 3, 2, 1, cheers. Again, on my count, in 3, 2, 1, cheers. This is the last one. I promise you, ladies and gentlemen, in 3, 2, 1, and cheers. That's it. Thank you so much once again. Before we jump on to the next session, I would like to remind all of our participants to fill out the attendance form by the link provided in the chat box, because the attendance form is needed for you to earn the certificate from the beginning until the very end of our session today. And after that, we are going to have a great discussions by Association of Veterinary Schools. By Asian Association of Veterinary Schools. Oh, this is why I'm so nervous. That's why I am a little bit lack of speaking, but I hope everything will gonna be okay. Ladies and gentlemen, the Asian Association of Veterinary Schools would like to provide a great discussion on globalization and challenges on veterinary education system. Professor Abdul Rahman Omar, joining us from Malaysia as AAVS president, would lead the discussion. So Professor Omar is a lecturer and a researcher from University Putra Malaysia. His main interest in research is about immunology and molecular virology. Without further ado, ladies and gentlemen, please welcome Professor Abdul Rahman Omar.
Thank you very much, Madam Chairperson. Bismillahirrahmanirrahim. Assalamualaikum, and a very good morning to all distinguished participants, delegates of ICAVESS. Welcome to the Asian Association Veterinary School session of ICAVESS session, where for this session So this session is jointly organized and supported by AAVS, the Faculty of Veterinary Medicine, University Gadjah Mada, and OIE. Let me first congratulate the Dean of the Faculty of Veterinary Medicine, University Gadjah Mada, Professor Dr. Siti Istrina, and the Chairman of ICAVESS, Dr. Widago, and his organizing committee for the success in organizing this seminar. As has been stated, I'll be chairing this session, where for this session we have invited two prominent speakers to discuss pertinent topics on veterinary education in Asia. Okay, so before we start, probably a few housekeeping matters. Each speaker will be given 25 to 30 minutes of presentation. Try to finish your presentation within 30 minutes. We will conduct the Q&A session at the end of the session after both speakers' presentation. Okay. Besides asking verbal questions during the Q&A session, participants also are encouraged. They can ask questions through the chat box. Okay. All right. To begin the session, our first speaker, Professor Dr. Muhammad Khair Bejo from the Faculty of Veterinary Medicine, University Putra Malaysia. A brief biodata about Professor Khair. He received his Doctor of Veterinary Medicine from University Putra Malaysia in 1985. Then he pursued his PhD in the field of veterinary pathology at the University of Liverpool, UK in 1990. Then he pursued his sabbatical training in the field of avian medicine and pathology at the University of Georgia, Athens, United States. He was the former Dean of Faculty of Veterinary Medicine, University of Putra Malaysia from 2012 to 2019. He is also the former President-elect and Treasurer of Asian Association of Veterinary Schools, Secretary for CSR, and also members of the Malaysian Veterinary Councils from 2012 to 2019. He has published over 200 papers in areas related to pathology and vaccine development. Currently, he holds the position as the Director of Putra Science Park, Putra Malaysia. He's also a Fellow at Agri Science Malaysia, Founding Fellows of Malaysian College of Veterinary Specialists, and currently the President of Malaysian College Veterinary Specialists. Professor Dr. Muhammad Khair, the floor is yours.
Rahman. Thank you, the Chairman, Professor Dr. Abraham Omar, Dean, Faculty of Veterinary Medicine, University Putra Malaysia, as well as the President-elect AAVS. Assalamualaikum warahmatullahi wabarakatuh, and very good morning to all my colleagues, ladies and gentlemen. First of all, I'd like to express my sincere thanks and appreciation to the ICAVESS and the organizing committee for inviting me and giving me an opportunity to share my thought on the globalization and challenges to veterans education in Asia in this conference. Globalization is a term generally used to describe an increased internalization of market for goods and services. High quality of veterinary education and efficient veterinary school, veterinary statutory body, veterinary profession, and veterinary association are so important to preserve the good governance and function of veterinary services toward global public good. A standard must be established and adhered, and it requires specialization. And indeed, it may need to re-engineer veterinary education itself to meet the global demand. In Asia, we have 51 countries with about 4.7 billion current population. We contribute equivalently about 60% of the total world population. Out of this, we have 22 members countries with about 63% have a veterinary school with a total of about 158 veterinary schools in Asia. And the size of veterinary school based on student intake varies as low as less than 50 and can be high as more than 200. When you look at the issue and challenges of veterinary education in Asia, we can see that there's quite a number of issues and challenges, such as social culture differences. There varies in terms of language, for example. People in the region speaking nearly 2,300 living languages. When we talk about culture, mindset, there's some variation. There are also differences in resources, operational and procedure. For example, in terms of the production, quality assurance, veterinary services, as well as national quality framework. There are also very strong in veterinary services in the region. There are also differences in the policy and governance, which involve a political commitment and regulation. Similarly, there are variations in distribution in terms of the workforce, gender, and practice type. We are also facing an issue with lack of educate incentive in terms of recognitions, opportunities, professional development, and perhaps compensation as well. Lack of access to border opportunity. And we are also having variation in trade policy. So these are some of the challenges and the issue that we have in the region. But as we are familiar, the veterinary profession deal with health and diseases in vertebrate, including human. In a global world today, the veterinary profession in the region may not be able to meet the need of the society and protect the broader carrier area. With the human population in the region continue to grow, and this growth will put increasing pressure to produce sufficient food in a sustainable way. There is a societal expectation that a professional approach is being taken to managing food security and food safety, as well as the environment and biodiversity. Thus, every veterinarian must accept the challenge of becoming and remaining competent, making a lifelong commitment to education. If the profession does not provide and serve that might reasonably be expected of it by society, it is certain that new or existing profession or discipline will fill the void. The profession must review critically that the present culture of profession modeled solely on the human medicine profession is no longer functional. It is a need to transform or perhaps re-engineer, if necessary, the veterinary profession from traditional clinical approach to one that fully serves our society. We need expertise. Specialized knowledge is the most important characteristic of the profession. As we are aware now, knowledge is now widely accessible and is becoming democratized. Technology will change the way the public get access to the professional expertise. There is tremendous increase in the technology like computers, genetic, nanotechnology, robotics, and artificial intelligence. Not forgetting the opportunity and challenges on the technological singularity. Veterinary education curricula and core curricula number credit hours are varies from veterinary school in the region. To ensure consistency and competency, veterinary graduate across country in the region curricula should be standardized, harmonized across at least a number of common and important subjects. As mentioned earlier, the viability in culture, language, art of communication, demand and requirement among member countries, background on the establishment of veterinary school, and financial constraint are among issues and challenges in harmonizing or standardizing veterinary education in the region. Veterinary education need to address the strength issue and challenges in new scenarios such as One Health, transmissible diseases, leadership, entrepreneurship, information technology, halal, food safety and security, as well as stakeholder expectation. Day one OIE competency or graduate competency, global recognition, program accreditation, animal ethics, and welfare. Indeed, re-engineering of veterinary education in the region may yet to consider critically in the near future by adopting education system such as combined substantive undergraduate tracking, compulsory internship, and designated licensure to address issues such as gender and shortage of veterinarians in livestock industry. We can see that we have more graduates working in companion animals. And again, we are not gender biased, but we can foresee that we have more female than male in the profession. When you look on the undergrad tracking, it may have some advantages and disadvantages, and this ranking may not able to retain considerably value in the breadth of the current veterinary degree with its opportunity of comparative study across the species. But we remember, veterinary learning does not end with graduation. In fact, learning has only just begun. It is necessary to continue throughout every veterinarian professional career. The need for continued professional development after graduation, an area of specialization such as postgraduate program, internship, residency, specialists, lifelong learning, and continuous professional development are already widely recognized by the existing accreditation body. Continued registration to practice veterinary medicine already depend on the achievement of the prescribed level of continued development. What is required is that we need it to be part of the mandate of global standard. The future of teaching and learning can be uniquely individual. We involve abstract thought, active learning, and higher order cognition. It might also involve personalized teaching, which is more customized, intense feedback, and safe, paced, and adaptive. Competency-based training and assessment focus on outcome, emphasize ability shift from time-based training to competency-based assessment. There's also a shift from faculty-centered teaching to student-centered learning. The outcome of the DVM program must be measured, analyzed, and considered to improve the program. Student achievement during the pre-clinical and clinical curriculum and after graduation must be included in the outcome assessment. What we like to see is a veterinary graduate must have the basic scientific knowledge, skill, and value to provide entry level of healthcare independently at the time of graduation. A minimum graduate must be competent in providing entry-level healthcare for a variety of animal species. Meeting of the day one OIE competency and meeting of what we call it as a graduate competency. Indeed, it is urgently need to develop a long-term strategy towards global recognition of the veterinary qualification. There is a social expectation that veterinarians everywhere will all have graduated at the same standard and have the same basic competency, at least meeting minimum standard of competency. This competency must be enable the profession to deliver the wide range of expertise and services required today in the foreseeable future. Currently, the dilemma and one of the major challenges that the profession faces is the significant disparity in the quality and recognition of veterinary education in region and around the world. A lead must be taken by the regional or global body to develop a long-term strategy towards global recognition of the veterinary accreditation. Currently, some regional blocks of accreditation operate around the world, and they encompass a significant profession of veterinary profession. The major accreditation bodies are based in North Africa, Europe, United Kingdom, and Australia, such as AHMA, ICAVESS, RCBS, and AAVC. There are some advantages of veterinary education, as veterinary school accredited could develop a well-educated veterinary workforce. But at the same time, we have to look that with this accreditation, we can promote continuous quality improvement of the veterinary school. It benefits the society in terms of quality of the services, benefit the student in terms of their investment in education, benefit the profession as we produce more competent graduates, and of course, it benefits the school, especially when we talk about continuous quality improvement. Okay, there's some constraints, which include particularly, perhaps, it will cost more on administrative burden to the faculty, a lot of paperwork, a lot of writing time to do writing self-study report, and many other things. But of course, another challenges or probably also the constraint is it will increase the budget of the school. The establishment of Association of Veterinary School, Asia Association of Veterinary School in year 2000, and Southeast Asia Veterinary School in year 2010 is partly aimed in harmonizing of veterinary school education in the region by setting a minimum requirement for the veterinary school towards the establishment of regional veterinary accreditation board. Currently, ICAVESS have 52 regular members with 3 associate members, and the first meeting of ICAVESS was held in 2001 in Seoul, Korea, Seoul National University. And the last face-to-face meeting was held in 2019 in Putrajaya, Malaysia. I think during this meeting, particularly on these three meetings, in 2008, 2009, and 2007, the meeting really focused on what we call it standardization as well as accreditation of veterinary education system in Asia. Because in year 2008, we are meeting in Seoul, Korea, with emphasis on standardization of veterinary education system in Asia. And the following year, 2009, same thing, follow up on the previous meeting, will really focus on accreditation and standardization of veterinary education system. And last meeting we have, again in Seoul, Korea, in 2007, instrument accreditation of veterinary school. The veterinary services is global good, public good, and in the front line in reducing poverty, food security, market access, animal welfare, protecting animal health, protecting public health, and animal production and food safety. A veterinary service in most of the ASEAN countries is a balance of medicine and production based on socio-economic status of the country. With the implementation of ICAVESS by ORE, good governance of animal health system based on the close public-private partnership is responsible of the government. In a scenario where if one country fails, it may endanger its neighboring country, the region, and continent, and potentially the entire global or planet. Veterinary statutory body in the region varies considerably from countries that have no form of regulatory to veterinary practice to those with high quality veterinary statutory body. In principle, we have at least three types of this VSB category, which is namely as independent, which is regulated by veterinary profession, and by veterinary. Together with effective veterinary statutory body are cornerstone of good veterinary governance. Ultimately, responsibility is vested in the veterinary statutory body that accredited veterinary school, set the standard examination for all new graduates, issue licenses, approve specialty board and veterinary school, and set a practice standard. In addition, ORE has recommended that the role of veterinary statutory body is to include in the regulation the activities of veterinary paraprofessional and ensuring these activities are ethical and high quality. Global and national veterinary associations are crucial to ensure quality veterinary education. In the era of globalization, veterinary associations in the region need to transform or re-engineer, meeting the need of the society and rapid change in veterinary profession, education, and services in line with international standards. The association should not progress like other non-professional associations, but as a professional association, we should learn from other well-established and successful professional associations globally to excel and provide significant services with high impact to veterinary profession and the society. We are so pleased and so happy to see that ORE has shown great interest in harmonizing veterinary program in the region and the world. At least ORE has established 4 ORE Global Conference. It was held in France in 2009, Lyon in 2011, Brazil 2013, and in Bangkok 2016. In addition, many meetings were conducted in the region and sub-regional by ORE office. ORE also has come up with guidelines such as Day 1 competency, mode core curriculum, and competency for veterinary paraprofessional. Not forgetting, ORE has started what we call it twin project since 2013. We can see that there's quite a number of successful or success story with regard of this OIE twin project. For example, though was funded by OIE, University of Minnesota and Chiang Mai University, Thailand. Those without OIE funding support, but be endorsed by OIE, for example, Mesa University and University of Paradenia, Sri Lanka. So these are a number of OIE twinning projects. But recently also, OIE also have focused on the accreditation, where we have established OIE Australian Veterinary Council and Veterinary Council of Thailand twinning project. And this project is focused, I could say, to establish what we call it a veterinary accreditation body in the Southeast Asia region or in the ASEAN country. The year 2020 until now has been a very challenging year to everyone throughout turbulence caused by COVID-19 pandemic. National lockdown, social distancing, wearing face masks constitute profound interference on individual freedom, social stability, and economic safety. However, despite grave challenges, we were able to steer teaching and learning activities into adopting to a new normal and able to use technology to our advantage in bringing value to our veterinary education. Online lectures, practical classes, clinical rotation, and conferences using such as webinar platform are now common to all of us. It benefits us, especially on the opportunity for the student to attend lecture delivered by many experts globally, while they are only stay at home. However, some feedback probably many of our hand-on practical classes, out campus, farm and clinical training yet to be postponed. During this COVID-19 pandemic also, there is significant improvement in terms of innovative approach for veterinary education. We use a new normal. ETL, supported blended learning, model-based system, online learning focusing on lecture videos, deliver drive link for record, and Google Meet and Zoom for synonymous session for practical cases are very common now. Some of disadvantages of feedback that we have to remember when you talk about the student also might have some issues and problems that we should need to consider. Particularly, they might have some constraint in financially. They might have some constraint in terms of the networking, for example, the internet itself might affect their study. Of course, they cannot do any much of extracurricular activities as well as physical activities. This may create a stress and probably can cause some issue in the mental of the student. We wish that our student is strong and they're able to cope with this COVID-19 pandemic successfully. In conclusion, the veterinary school, veterinary statutory body, veterinary services, and veterinary association in collaboration with and support by national, regional, and global organizations such as OIE play a vital role for quality veterinary education to ensure the success of veterinarian to provide holistic solution to changing global challenges. The region is prepared for challenges of globalization to veterinary education, though there's a big horizon to explore and take more time before it can be successfully achieved and become a fully reality. There's a lot of opportunities for veterinarians to provide holistic solution to challenging global challenges. COVID-19 pandemic has increased the use of technologies and innovative teaching approaches, a new norm, and perhaps will be the future of teaching and learning practices. Lesson learned from COVID-19 also, like COVID-19 is one of the worst pandemic, but please remember, it is not the first and possibly not the last. It is indeed one of the most critical and high-risk challenges to veterinary education and its ecosystem globally. We need special attention, strategies, and quick action for a short and long-term solution. With that, thank you very much.
Thank you very much, Professor Dr. Muhammad Hair, for a very insightful presentation, sharing some views on the key challenges in veterinary education in Asia in this area of globalization. I already noticed some questions trickle in the chat box. I believe more will come. We will compile all those questions and we shall discuss during the Q&A session. Okay. Our second speaker for the veterinary education in Asia session, together with us, we have Professor Pang Dong-Ru from College of Veterinary Medicine, Seoul National University. So his presentation title will be on veterinary education and accreditation of veterinary schools in Asia. A brief biodata about Professor Pang. He's a professor of veterinary pharmacology at College of Veterinary Medicine, Seoul National University. He received his DVM in 1979, PhD from Iowa State University, and then postdoctoral training in Yale University. He joined Seoul National University as a lecturer in 1991. Professor Ru holds key positions, including Dean of College of Veterinary Medicine, Seoul National University from 2011 to 2015, President of ICAVESS, President of Korean Association of Veterinary Medical College, and Korean Society of Veterinary Medical Education. He has also served on the OIE ad hoc group on veterinary education and sat as board of the directors for Accreditation Board of Veterinary Education in Korea and on the Education Committee, Korean Association of Veterinary Medical Colleges, for the development of a competency-based veterinary education and national model of Day 1 competency in Korea. Professor Ru has published more than 170 research papers in neuroscience and veterinary pharmacology and was director of BRAIN Korea 21 program from 2006-2011. He also the editor-in-chief of Journal of Veterinary Science and Journal of Acupuncture and Maritime Science. Please welcome Professor Pang Dong-Ru.
Thank you, Dr. Omar. Also, I would like to thank you in me this chance to share my ideas. Let me check the first point. Now, okay. The title is Veterinary Education and Accreditation of Veterinary Schools in Asia. Also, lots of my introductory material is already provided by Dr. Hale Bejo. Thank you. You know what is accreditation. In general, accreditation is a type of quality assurance improvement process, and usually external persons or organizer visit and then check whether they meet the standard or not. So in other sense, as say Dr. Gallant, advantages of accreditation is providing eligibility for licensing them. And the second one I focus is the assurance that equivalent standards are used to measure day 1 competencies of the graduates. So personally, I think that accreditation of veterinary schools is like an external recognition of the curricula that is designed to equip their students with day 1 competencies. Of course, day one competencies should be based on local and global societies meant. So I would like to say that accreditation and curriculum for day one competency is the different side of the same entity. Because of this, I like to first introduce or briefly summarize, actually it's not brief, OI day one competence and AVMC competence-based veterinary education, then go back to accreditation in Asian countries, and then finally I will summarize my suggestions in few slides. This slide shows that outcome-based education. So the outcome or result of education is determined or assessed by educational outcomes, actually student performance. Then this feedback to what kind of student we need to recruit, or what kind of support, how to teach, what to teach. So outcomes this indicates that the pedagogy in current days is the outcomes matters in medical education. And competence-based medical education is a part of this outcomes-based education. Initially proposed by WHO in 1978, the current definition of competence can go like this: an observable ability of a health professional related to a specific activity that integrates knowledge, skills, values, attitudes. Competencies are observable, can be measured, can be assessed. And probably you have heard a lot, day one competencies. Day one means right after the graduation or entry to the society. So some people say entry-level competencies or graduation competencies. Sometimes people say that minimum or core competencies, because this competency is a requirement for every student. The student who shows very low academic performance, still that student fulfill this requirement. Because of that, we say sometimes minimum or core competencies. So one of the familiar competence we have is OIE, day one competencies. This is already much discussed or introduced by Dr. Hare. So OIE held that 4 consecutive and they actually identified veterinary services as a global public good. And then in 2012, 2013, they developed and published a recommendation for day one competence, and the next year, core curriculum guidelines. And since then, they are continuously promoting, as Dr. Hale said, a lot of activity was going on up to today. Here is OI front face. Their mission is reducing poverty, securing food security, increasing market access, increasing animal production and food safety. So for this activity, the OI recognized that quality veterinary education is one of the cornerstones of OI's mission, good veterinary governance. In their day one competencies, probably all you know very well, so I will just go briefly. So basic, general, basic, specific, and advanced competencies. And basic competencies are really related to the critical competencies written in OI terrestrial code. And Actually, advanced competencies can be obtained after graduation, like the on-the-job training. And in case of general specific competencies, there are 11 competencies from epidemiology to communication skills. Each competency has learning objectives, sometimes 2, sometimes 4, 6. And each competence has the brief definition, like emerging disease, new infection, known and previously unrecognized, something like this. And also definition about re-emerging disease. They add learning objectives, define emerging disease, and provide contemporary examples. Like there is 4 learning objectives in this case. So in case of advanced competencies, there are 8 competencies, and each competence has the related objectives from research and administration. So total, there are 11 specific competencies and related 39 learning objectives, and 8 advanced competencies and 33 learning objectives, total 72 learning objectives. Probably you have been trying to incorporate these learning objectives in your curriculum. And then OIE in second global conference, they also suggested minimum core curriculum, or like in these documents, and to promote for that implementation of the day 1 competencies, they prepared and shared these core curriculums. So total 20 core courses or programs. But the OIE competencies, which is focused on the mission of OIE. But if we think about the veterinarian, there are many other things that veterinarians can do or have to do. Those are probably, we need much larger or broad, comprehensive competencies. So these competencies include basic sciences and skills, clinical sciences, skills and performance, and non-technical competencies. And one of the good example and most up-to-date example is AAVMC Day 1 competencies. So here AAVMC is Association of American Veterinary Medical Colleges. So for competence-based veterinary education, CPVE, AAVMC developed a competency framework like competencies, sub-competencies like OIE, but they have additional one, interest or professional activity and milestones. I will introduce briefly about this. So to prepare this document, AAVMC formed a working group, yearly experts in Europe, Utrecht University and Royal Veterinary Colleges. They are Harold Bock and Stephen May is with well-known educators in veterinary medicine. They worked for 3 years and then published 3 documents. So one is CBVE framework. This is really similar to the Day 1 competencies of OIE. And the another one is interest or professional activities. And the third one is milestones. So in the CVV framework, there are 9 competence domains here. And so the first domain is clinical reasoning and decision making, individual animal population, public health, communication, collaboration, professionalism, financial scholarship. And in these domains, so OIE 대연 competencies are spread here. And their domain structure or their competency frame structure is like this. So domain 1, clinical reasoning and decision making. First they have description, what this domain deals. And then they have the competencies, individual competencies like gather, sense, or assimilate relevant information about animals. Can you imagine what to do under this title? Some knows well, but others may not. So they further provide these illustrative sub-competencies. So there is a 4. This is really something like the learning objectives, but in details probably a little different. Anyway, so this kind of sub-competencies is not intended to be all encompassing or comprehensive. So you can add more according to your situation. So the full content of domain 1 of CVV framework is here. This is, you can obtain this paper here. And number 4 domain, public health, you can say that here, graduate respond to issues at the interface of animals, humans, and environment. Can you see this is really the 1S. So 1S component all included here. Well, this is all based on the earlier competence-based medical education. So CVME is the base of the CVVE, just I introduced. But CVME is well accepted, but is too analytical and too abstract, sometimes difficult to grasp as cannot touch. So in during 2010, people started to think we need something other, something related to work rather than individual competencies. Dr. Ten Cate in Netherlands proposed, and now during up to the 2015, many medical schools adopted this one. So what is the EPA? Entrustable professional activity, a duty or activity in the clinical setting, maybe workplace, we can say, that may be delegated to a learner by their supervisor. He or she has demonstrated sufficient competence to perform this task within minimal supervision. So this indicates that there is a patient in the hospital, but you have something urgent at home. So can you ask your associate, please handle this patient, then I can solve my problem at home and will come back. You can trust, or you can entrust your associate or your student in the hospital. That is the state or competence state targeted in this EPA idea. So EPA describes an activity in the workplace that can be used as an opportunity for assessment of the learner. So the workplace not necessarily be the hospital, could be the farms, could be wild field. So these the terms. So what's the difference then EPA and competence? Then EPA, as we mentioned, units of work tasks that must be done at the workplace. And competencies, this is the qualities of individuals. I can possess the qualities. So here one can possess competencies, a person can have that competencies, but a person cannot have the diagnosis or the surgery, because EPA is the work descriptors like discharge the patient or lead family meeting or explain or counsel the patient. But the competence, you may see that knowledge, skills, attitude, and values. So this up to now I just introduced that what is EPA. So now I will introduce the EPA of AAVMC. They have 8 core EPAs. There are many reasons why they set this way. And you can see the reasons here in this paper. So first one is gather a history, perform an examination, and create a prioritized differential diagnosis list, develop diagnosis plan, interpret result. And here is the surgery. Here's anesthesia. The another one is, the last one is preventive medicine. Actually, the 8 is more focused on the public health. And this 8 core EPAs also covers the animals, individual animals, populations, and different species. So their document, this shows the EPA structure. So EPA 2, the title is develop a diagnostic plan and interpret the results. And there is description of the activity. What is EPA? And what has the EPA has the, what kind of meaning EPA has, or some other additional comments. And then they connect the competence domains here. There are 2 major or most relevant domains, 1 and 5 domains, 1 and 5 domains, tips domains. And so 1.2 or 5.1 is individual competencies mentioned here. So in this here, EPA is mapped to 9 domains of competence. And here in elements identifies the specific competencies within the domain that make up the activities of the EPA 2 in this case. So I illustrate a different way. Of course, I got this one from the AAVMC conference. This is not my own slide. So there is EPA 1. It's a different, at least 5 relevant domains of the competence. And each domain, there are specific competencies related to this. So you can see that how EPA is structured or mapped with competence framework. So this is the whole table of cross mapping of core EPAs 1 to 9 and 8, and domains of competence from 1 to 9. Well, of course, you can get this one from the paper just I showed before. So now I wanted to discuss or introduce the milestones. Milestones is really the rating scales of interestment or rating scales of interestment. So milestones in another way, a level of achievement in each competence. So in a specific competence, or student can be novice or advanced beginner or competent or proficient. So usually the competent, this level is the level of what day one or graduation. And this one also we can express this way. So a student start from this one and then with the years getting competent or proficient. This is on one example that competence 1.1 gathers and assimilates relevant information about animals. This competence is related to EPA's 1247. And students should have this level for graduation here, performed through physical exams in logical and fluid sequence, identifies and documents most abnormal physical exam findings, including subtle findings, and interpretation of result adequately and supports the plan. So they specified what is the competent level. So again, this is the other illustration. So EPA 1 has relevant domains, and each domain has specific competencies. And a student can be fluent from the beginning in thinking or collaboration from the freshman, but probably not fluent in diagnosis as a professional activity. And here I said EPA, and this one, the milestone is one competency. You can combine all these milestone or the achievement levels in level EPA 1, EPA 2, EPA 3, and whole EPA, for example, EPA 1 of the first or certain student can be said not ready to trust or trust with no guidance. So I can give the bench to a student and ask, gather history and perform examination, create a priority differential diagnosis list, and then go back and have the dinner and come back and see what's your performance. But if the students are not trusted, you cannot go home for dinner. So up to this part, I just introduced the current major Day 1 competencies. Now I will go back or briefly show that progress in veterinary education and accreditation discussed in ICAVESS. So this is on my previous slide. Maybe more than this, VEs, veterinary schools, more than 128 schools. But anyway, this is the result of observation in 2016 from OI website. Some countries have only one, some countries have many veterinary schools. Of course, this is depend on the countries and the culture. Here, so in this pie graph, I showed that who approved the curriculum. In 12 countries, university approved. And also to many other countries, the curriculum is finally approved by the people or organizations other than university. And in this right side pie graph, the accreditation is provided or performed by Ministry of Education or veterinary statutory body. This is more than half. And about 1/3 of the countries, the accreditation system is not clear. And also we may see this data. This is from the OIE's paper, Dr. Urabe here, and she is also helping us a lot. Thank you. I like to thank you here. So here, majority of veterinarians are working in companion animals in 9 countries, but in some countries, majority of veterinarians are working in government or in food processing industry or livestock industry. So in this case, their own competencies should be focused. Their major activities in the society. So already I mentioned, but I like to summarize here, about 1/3 of veterinary schools in Asia are not under a quality assurance by accreditation system. And the curriculum change may take a longer time in many countries because the decision is made by others outside the university. This is what I guess. And the major role of the veterinarians vary widely among countries. Now I turned on to ICAVESS activity, which is well introduced by Dr. Bejo. So we had 18 annual meetings and 1 workshop for veterinary education in 2014. Here is the picture under the leadership of Dr. Siddiq. I feel okay. She was passed away. Okay, I will continue. This meeting was September 7 to 9, and the objective was here provide, of course, standards and prepare it, and then provide the ground for accreditation system. And based on this, in the 2015 ICAVESS meeting in Thailand, there is a ICAVESS working group was formed, and Dr. Siddiq was the chair, and the activity was reported the next 2 years. But when I look the activity of the ICAVESS on the education, there are some things in each meetings. So now this is the 18th meeting, but there's little accumulation. In 2021, at least if we combine all these, at least there must be something like this. Do you agree? Why? Why we don't have this kind of accumulation? I think personally, there are many reasons, but the primary reason is a lack of a channel allowing a continuous communication. Deans participated. After their term, what they talked, what they decided is not followed by the next deans or next presidents of ICAVESS. Now, I like to say a little more that OIE. So I like to say that OIE has been the game changer in veterinary education in Asia over the last 10 years. Already mentioned, there is a guidelines, recommendations, supported training programs, a lot of supporting, continuous long-term support, not only in Asia, in Africa and South America too, and workshops well introduced by Dr. Ye. And OIE set standards, but not involved in accreditation of individual veterinary schools. in some veterinary schools are looking for accreditation from the established accreditation bodies outside Asia, from AVM and COE, Council of Education, or EVE, or AVVC in Australasia. And some are success, some get accreditation, some are still preparing. But I think this is a good move, whether they got accreditation or not. The final goal, eventual goal, is to improve their quality of education. So this is a good move, I think. So now I will conclude my presentation, suggesting few strategies. So the first suggestion is, let's establish a society of veterinary educators. So to form an Asian community of practice for veterinary education and maintain communications and contact among members. And those members can be individual faculty members or experts in the accreditation bodies of each country. And then we need to identify proper day one competencies according to local demands. And then prepare suitable curriculum or educational program. This curriculum or educational program, also we can say different way, own CBV or EPA program. Then this is the kind of blue script, blueprint for the building. Then we can design what kind of system we can make, maybe. First, we have to agree what's the advantage or benefits of adopting that accreditation system, and then prepare accreditation standard. And the third stage, then we work on implementation of accreditation system. There may be hurdles probably within your school or within your country. In the school, the different other faculty members, and in the country, there is a law that prevents or delay educational reform and accreditation. And then we identify these hurdles and work together to overcome them. And then, again, we have to maintain continuous improvement. First, the educators should study, research on the veterinary education, and should know the changes local and global societies or ecosystems in these days. So thank you very much for your attention. I hope my suggestion can be shared and discussed further. Thank you very much.
Thank you very much, Professor Ru, for a very good presentation in summarizing the journey took by Seoul National University to achieve accreditation by AVMA, and of course, some of the recommendation suggestion probably put up to ICAVESS in terms of how we can actually move forward in terms of all the previous effort we have put in with all the workshops, seminars, in compiling the needs for us to improve the veterinary education in Asia, and once we so some of the strategies that we have in mind. And we have seen also a lot of some question coming in through the chat box, and also at the same time for the Q&A session, it's going to be an open session. Participants are most welcome to express their question verbally, and I also will go through some of the questions that was being shared in the chat box. So basically, Q&A session, I open the session to Q&A. If any among the members have any specific questions, are most welcome to give their views. And okay, all right. So is there any question, or shall I start with the question that from the chat box?
Oh,
Yes, please, Professor Hori. Yes.
Oh, okay. Thank you very much, Dr. Bajo and Dr. Liu, for your valuable lecture. I was thinking so many things as a vice president. So this is a comment from me. So I think recently livestock infectious diseases such as African swine fever are now widespread in the world. In addition, infectious diseases that were originally transmitted only in animals are becoming more infectious to humans due to mutation in the virus, and are becoming more and more harmful to human health. So in today's world of globalization, so SARS-CoV-2 has caused a pandemic all over the world, including Asian countries. So these mean that infectious diseases of various animals affect food security and harm human health. I think the term zoonosis is no longer needed. So now is the time for veterinary medicine to play an active role in the world to protect food safety and human health, I believe. So by the way, so as you suggested, so AAVS has more than 50 universities as regular members and associate members. So it is one of the candidate groups as an organization to create accreditation body for veterinary education in Asia, I think. So, but unfortunately, I don't think the current AAVS is realistic, as Dr. Liu has been shown. So until now, so AAVS has only been a salon-like place for the information exchange for dean of the each veterinary college. So, but recently we have to change. We have to change new AAVS meeting. So, so I'm now president. So I want to say message to yours. So first of all, we must strive for what kind of activities and the services we can provide to you as AAVS. First of all, with the help of OIE. So I think it is important to carry out some kind of activities centered on AAVS to share knowledge and skills on epidemic diagnosis and the treatment of infectious diseases spreading in Asian country with all member universities. So I will, if that happens, so I think it will be the core of the Asian veterinary educational accreditation body. So we have to start to do something. We have to do activity for making our own standard Asian veterinary education. Thank you.
Thank you very much, Professor Hori, for very much true. Looking into the current scenario with a lot of problems on African swine fever, we have even influenza, now we have COVID-19, more and more transboundaries diseases and also zoonotic diseases are actually of a significant threat to the animal industries, including to the human. And AAVS is actually a very good platform for us to organize activities looking into how we can strengthen playing our role, especially in diagnosis and controlling some of these problems. I believe we can organize more activity on this, and we are hoping that once this COVID-19 challenges are getting less a problem, I think more meetings, physical meetings, workshops, and everything can be organized very much. Right. Okay. coming back into some of the questions, a lot of questions been thrown in here, and if I can look at it, of course, one of the key challenges we have here, being a veterinary school in Asia, we are so diverse in terms of challenges. In fact, in terms of our animal diversity, in terms of where our graduates are working, is also pretty much very much different. So one of the key challenge, one of the questions that I can look into here is how can we strike a balance, try to harmonize the standard that we have, that we must comply in our veterinary school, and this by using different approach so that we can actually address the uniqueness of our challenges or concern, at the same time try to have a very common standard. And of course, probably Professor Pang Dong have indicated probably one platform is the OIE. Day one competency will be a good stage for us to launch or try to harmonize these particular variations or deficiencies that seems to be quite wide among all the different vet schools. Perhaps Professor Ru, you want to highlight a bit more? I believe the day one competencies definition or interpretation among all the various vet schools so probably pretty much different. Looking into because if you look at it, you look into it, whether is it focusing on a livestock problem or is it on a companion animals. I think this is something again all of us need to be on the same page in terms of understanding or defining this OIE day one competencies.
Can I say?
Yes, please.
OIE day one competency are probably the common all the country, all the schools. But other proposed by ICAVESS could be optional to some countries, and some countries should prepare their own competency or competency because the social needs are different, and any accreditation body should reflect that kind of the difference in among the countries.
Okay, yes, as been stated by Professor regarding the day one competencies, and if I can look at it here, all right, thank you. There's also a question regarding yes, resources are very important. Well, all of us, of course, when we to convince our university management to get more funding, especially to continue improving our program and eventually to get accreditation is very much challenging. And over here, looking into a suggestion, looking into adopt the practice in Europe that each university is measured by its own standard. Well, this is something that probably some suggestion from Eve. Probably they are using different model in going for accreditation. Perhaps any of the two speakers have some experience commenting on this Eve accreditation standard. Probably anyone? Yeah, Profi? Yes.
Good afternoon, Prof. Rahman. Probably not really direct to Eve, but I think what is more important with regard of our region, maybe I want to respond a bit on the Prof. Hori mentioned just now. I think the first thing is that for the past many years, we are working with OIE, really focusing on veterinary education. Okay, so that is something that we really appreciate the initiative of OIE to ensure quality veterinary education in the region. For the future, I think looking at all the pandemic that we have, I think one of the important things we look, maybe we start to work with OIE also on the One Health, and it really focus on One Health. I think there's many organizations or many entities have worked on One Health. For example, when you talk about the Southeast Asia, we have South Asia One Health UC network, really fully funded by USA, and that was established since 2011 and still ongoing. And I do understand also in many regions, I think even in Japan and Korea, you have also those of One Health interest. You know? So I could say that probably apart from veterinary education, we should focus on One Health and with the support of OIE, because OIE is very strong members of tripartite, WHO, the FAO, and OIE. So that perhaps can give an impact to the pandemic. Yeah. So when we refer to veterinary education itself, I mentioned earlier, OIE have contributed significantly. Of course, there's some impact, but probably we would like to see more impact, what I call it, especially to AAVS. I could see that the impact for SIAPSA is there. I think currently with twin project between OIE and ABBC and the Thailand Veterinary Council, they really work hard and they come out maybe with standard or minimum standard that to be complied or suggested to the veterinary school. I think what's more important, there must be a group that should work for the minimum standard. Where our direction, if we thought about 11 or 12 standard being established by certain accreditation body, we might have looked at carefully. And then with that, we determine what is the need of our region. I do understand that we have so many countries, 51 countries, so many veterinary schools. We have big challenges, but I always consistent. We have to start somewhere. And where to start somewhere is come up with the minimum standard. And from there, we improve the standard from time to time, meeting the requirement of each country, because each country has different requirement, but we can have a range. For example, we can put certain range. We have a compulsory basic requirement. And maybe those basic requirement, as Professor Pandong mentioned, maybe they want OIE competency, but people also talk about they want graduate competency. So we put a minimum requirement, and then some varies among country to country. And I think we should start somewhere. I'm sorry, I'm always consistent about this, and I think we should work it out. And initiative have been done previously. And when you look at if also, if it's more flexi, isn't it? Although there's some limitation and so on and so forth, you compare with AHMA or even ABBC. So we can learn from that. And from there, we can use it as our benchmark to establish our own. Because ours is very unique. There's a lot of challenges. There are a lot of variation. And many of the vet schools in the region try to have what we call the accreditation from these bodies. But of course, they are different. For example, in terms of public health, for example, there's some degree of variation in terms of focus. Then when you talk about caseload, for example, there's different variation among country to country, in which if we have our own regional, so we can fit for each country with some variation, depending on the interest of the country. But at the end of the day, we have something to refer to a standard for us. Thank you very much.
Thank you, Dato' He. Very strong believer in AVA. We should come up with our own minimum standard. I think looking into all the workshop to follow up in terms of is it possible for us to come up with minimum standards, addressing all the different needs, at the same time addressing all the different needs of all the various, strengthening up the component of One Health. There was also a question regarding how strong is the component of One Health in our veterinary curriculum. And I believe the component of One Health is actually embedded in some of the courses that are available now. And probably the percentage or the loading will be probably varies from one universities to university. This is something I don't know. I mean, perhaps probably we need to also to look at is there a need for us to have a dedicated course on One Health, or is it whether the current emphasis on One Health, whether in public health or on zoonosis, is actually something is sufficient enough. This is something probably definitely we will look into it as we progress and have more meetings and discussion. Okay, right. I think we can. The information I got from the organizing committee, few more minutes. Because deans, representatives from all the various vet schools in Asia to discuss very important topics on veterinary educations and accreditation. If I can look at it here again, there's one question regarding there are some vet schools that actually they have been accredited by national bodies, and where they don't have proper veterinary statutory bodies. So there's also suggestion that from all the various example of VSB from different countries, we should share informations regarding how we can actually learn from each other in terms of the veterinary statutory bodies that are from one country to another country. I think basically that's pretty much true. I think all our veterinary statutory bodies, we have different levels of our engagement in terms of how we go about addressing our veterinary program. This is something that very much true. What we notice whenever we want to improve the quality of veterinary education, then we need to have a very strong collaboration between the veterinary education establishment with the veterinary services, with the veterinary association, including the veterinary councils, the national veterinary council. This is something that very much need to be given emphasis over and over again. Right. If I can look here again, some of the questions. Right. Okay. Okay. I believe many of these questions we will take into note, and definitely there are going to be a lot of rooms for us to discuss further, probably in our coming up ICAVESS discussions and meetings. Okay, all right. So basically, we will take note all these questions, and we will look into it as we come up. So any more questions from the participants? I think we have listened to a very insightful presentation from both our speakers, Professor Dato' Muhammad Heer and also Professor Pang Dongru, for giving a very insightful presentation, sharing your views and ideas, very important topics on how veterinary schools in Asia from a very diverse backgrounds, deficiencies and strength, how we can actually should move forward in a one voice and one effort, so that we can continually improve the veterinary education that we have in our respective universities. The key challenge here, the key important point is to improve our curriculum. Once we have established the system, the standard that we need to do, and we need to monitor and improve it, and eventually we should able to get the tools to evaluate it and eventually to go for accreditation. That seems to be the journey that all of us need to explore in order to ensure that our program, veterinary program, is actually relevant, meeting the challenges and meeting the demands that we have in mind. Okay, all right. I think is there any more questions from? If there's no more question from the participants, before I end, let us express our appreciation to both our speakers, Professor Tom O'Hare and also Professor Pang Dongru for an excellent presentation. And on the behalf of the chairman of this session, thank you. And we will definitely look into all the questions that have been raised, and this open up a lot of opportunities for ICAVESS to work together and to organize meetings. Definitely, we will meet again very much probably during our meeting that been planned by our President probably. Okay, with that, all the best. Thank you very much for the invitation. Stay safe. I will pass back the session back to the organizing committee. Thank you.
Thank you.
Bye-bye for all.
Thank you.
Thank you.
Thank you, Omar.
Thank you.
Thank you so much, Professor Omar. Thank you so much, Professor Omar, and then Professor Bejo, and also Dr. Pantong Diu, and also Professor Hori. Thank you so much. And after this, we are going to have another virtual photo session. Since there are more than 200 participants joining us, I will divide this session into two parts. The first one, I'd like to invite all of our invited speakers and also all the deans from Faculty of Veterinary Medicines around Indonesia. And I'd also like to invite all of ICAVESS members to turn on the camera. Are you all ready? On my count. Professor Deku, may I ask you to turn on your camera, please? Okay, on my count, ladies and gentlemen, let's start. In 3, 2, 1, cheers. For the next page, another 3, 2, 1, cheers. Okay, the last one. In 3, 2, 1, cheers. Thank you very much, all our invited speakers and also the distinguished guests. Up next, we are going to have the virtual voter sessions with all of our participants. So that is why I'm asking you to turn on your camera, please, ladies and gentlemen. Are you ready? On my count. In 3, 2, 1, cheers. Another one. In 3, 2, 1, cheers. Once again, in 3, 2, 1, cheers. One more time for all of you, ladies and gentlemen, all of the participants, to please turn on your camera. Our host already invite you to turning on your camera. Are you ready? Another session for all of participants. Okay, in 3, 2, 1, cheers. Let's move on to the next page. In 3, 2, 1, cheers. Again, in 3, 2, 1, another cheers. Thank you so much for your patience, ladies and gentlemen. Okay, ladies and gentlemen, after this, we are going to enjoy the break session for about 45 minutes. However, I'd like to invite all of you to join the parallel session at 12:45 Jakarta time. Once again, at 12:45 Jakarta time, through the link provided in your email. And if you haven't filled out the attendance form, please click the link provided on your chat box. With this, I'd like to thank all of you, all of our invited speakers, our distinguished guests, and all of our participants. Please enjoy your discussion today. Stay healthy and stay positive. I'll see you tomorrow. Have a wonderful day, everyone. Thank you.
Universitas Gadjah Mada, or UGM, is a public university renowned as the People's University, which strives for and puts forward the interests of the citizens and nation of Indonesia. As a higher education institution that has long been recognized by the society, we are proud of our timeless and tireless dedication to the nation. By cultivating, implementing, and leveraging education, research, and community services, which constitute the salient objectives of Indonesian higher education, or Tridharma, we remain committed to incessantly promoting the values of Pancasila. We exert the utmost effort to protect Indonesia's sovereignty in accordance with Pancasila ideology and the 1945 Constitution. As a world-class university, UGM is devoted to the interests of the nation and humanity. We conduct research, provide education, and deliver community services by enhancing knowledge that benefits the community, the nation, and the world. We have been learning from one another and get stronger together since we believe that knowledge is not useful unless it is shared with others. The learning process will never cease developing. At UGM, we learn by continuously exploring sciences, learning from history, observing the present, and researching for the future. We always work hard and never give up. The learning and research processes at UGM continue to run together and explore state-of-the-art innovations. To create a convenient, environmentally friendly, and pollution-free green campus, UGM has built a wisdom park that substantiates the teaching and research purposes. Furthermore, UGM has established and run a science technopark as an embodiment of its capability of inventing and commercializing a myriad of innovative health products, ranging from medical devices, herbal medicines, and healthy food.
There are now more than 118,000 cases in 114 countries, and 4,291 people have lost their lives.
2020 has been a tough year for anyone, and it is no exception for UGM. Campus activities are abruptly halted, student activities on campus are annulled, and everyone is forced to work, worship, and study from home. Lecturers have no choice but to learn how to teach effectively using the information and communication technology, or ICT. Learning and research on technology are strongly supported at UGM, and in fact, we have conducted various cutting-edge technological research. Community Service, or KKN, is one of the most important courses to complete the study program, which will be undergone by University of Gadjah Mada, or UGM, in 2020 while adapting online courses system. Supported by the Ministry of Education, UGM will continue its service within the society across Indonesia during the pandemic issue. Lecturers and researchers at UGM continue carrying out basic and applied research in many realms. For instance, studies in the health sector to cope with COVID-19 are being encouraged and accelerated as a form of UGM's commitment to mitigating disaster using scientific methods. One of the most innovative inventions regarding to mitigating the COVID-19 is Genos, a screening tool or early detector of patients exposed to COVID-19, which is able to identify the symptom through exhale within 80 seconds. Genos is highly reliable. It has a number of advantages as a COVID-19 fast detection tool. Its sensor is able to detect more than thousands of patients in the long term, while providing relatively fast results, non-invasive and affordable. In addition, its usage also applies non-rebreathing mask and disposable HEPA filter. Those challenges somehow direct or even force us to innovate and modify approaches. Success is a journey which necessitates smart work through learning, studying, and sacrificing. Despite all that, most of all, we love what we do.
The Faculty of Veterinary Medicine of Universitas Gadjah Mada was established in 1946. It was started from the Bogor Veterinary Medicine College. In 1947, a parallel class was opened in Klaten. Faculty of Veterinary Medicine has 4 programs: Veterinary Medicine Study Program, Veterinary Profession Education Study Program, Master of Veterinary Science Study Program, and Doctor of Veterinary Science Study Program. Various tasks and expertises of veterinarian are learned in 12 departments. Each department is equipped with advanced laboratories and modern research tools. The 12 departments are Department of Anatomy, Department of Biochemistry, Department of Pharmacology, Department of Physiology, Department of Surgery and Radiology, Department of Internal Medicine, Department of Veterinary Public Health, Department of Microbiology, Department of Parasitology, Department of Pathology, Department of Clinical Pathology, Department of Reproduction, Obstetrics and Gynecology. Besides an advanced laboratories inside all of the departments, this faculty is also supported with animal health education and training unit, used by university students and veterinarians for the sustainable learning activities. This unit is called UP2KH, which manage several livestock such as cows, sheep, goats, and horse. Various achievements have been obtained by the Solari Society, such as the Regan tester kit for chicken carcass. FVM lecturers discovered this tester kit. It has been patented under the name Durante and has been widely used by the community. Faculty of Veterinary Medicine lecturers regularly write and publish books related to the veterinary medicine world that can be used for readers in the animal husbandry, biology, and statistics. The university students has also obtained several achievements in the research, arts, and sports field, and also national and international organizations. Some collaboration in the national level has been made. FVM UGM is the member of Indonesia Veterinary School Association. An MOU has also been signed between the government and private sectors for the purpose of research and community service. As for the international level, FVM UGM has also collaborations with foreign universities from Australia, Thailand, and Japan for research and education and other abroad universities. Furthermore, FVM UGM accepts international students from abroad, such as from Malaysia. Work principles, manusia serve mankind through animal kingdom.
Yogyakarta is known as a city of education in Indonesia. Such name is close to the existence of the oldest state university in Indonesia, Universitas Gadjah Mada.
Profesor Suparwi Animal Hospital, Faculty of Veterinary Medicine, Universitas Gadjah Mada, is one of Indonesia's leading veterinary hospital facility that is based in Yogyakarta. This hospital is also integrated with Faculty of Veterinary Medicine, Universitas Gadjah Mada, as a teaching hospital for the students to implement their comprehensive studies in real cases. As an educational hospital, our practices implement the three principles of higher education, including education, research, and community services. Our vision is to be a superior, independent, and dignified world-class animal hospital and being imbued with Pancasila serving the interest and prosperity of the nation. Our mission is carrying out learning and service in the field of animal health to improve human well-being based on research. In addition, increasing education, research, and community service activity in the world-class animal health sector with population identity and building Indonesian social culture.
Our team of caring and highly qualified veterinary staffs treat over 5,000 animals each year, supported by dedicated veterinary nurse administrative staff. Our veterinary care goes hand in hand with a natural love for animals. This is demonstrated to each animal brought in, whether for a simple checkup or a primary medical treatment. Furthermore, they love animals as much as you do, and it is shown in everything they do. In addition, Professor Supari Edible Hospital also provides house call service. Among them, medical service and grooming can be done outside the clinic, according to the agreement. In particular, we provide a laboratory, hematology with full blood work panels, radiography, another laboratory service with the results on the same day, so your pet doesn't have to wait for a diagnosis.
For this reason, we are committed to helping you provide your special family member with excellent care because they deserve a long, healthy, and happy life.
Universitas Gadjah Mada is an Indonesian public Ivy League research university and is the oldest and largest institution of higher education in Indonesia. UGM has been considered to be one of the most prestigious universities in Indonesia. Faculty of Veterinary Medicine UGM has been developing knowledge and science since the early establishment of the university back in 1946. This started since it was still unified with Bogor Veterinary Medicine College, until a parallel class was opened in Klaten in 1947, which became the pioneer and forerunner of Faculty of Veterinary Medicine UGM. Currently, Faculty of Veterinary Medicine has 4 programs: Veterinary Medicine Study Program, Veterinary Profession Education Study Program, Master of Veterinary Science Study Program, and Doctor of Veterinary Science Study Program.
Fakultas Kedokteran Hewan Universitas Gadjah Mada lahir sebagai bagian perjuangan Negara Kesatuan Republik Indonesia yang memiliki cita-cita mencerdaskan kehidupan bangsa sekaligus berperan dalam perdamaian dunia. Maka Fakultas Kedokteran Hewan UGM memiliki visi menjadi pusat unggulan pendidikan tinggi kedokteran hewan bertaraf internasional yang dilandasi jiwa Pancasila. Visi tersebut diwujudkan dalam proses pendidikan yang tetap menjunjung tinggi kearifan lokal, namun tetap mengikuti perkembangan teknologi kedokteran hewan secara global di era revolusi industri saat ini. Fakultas Kedokteran Hewan UGM mengembangkan riset dengan memanfaatkan kemajuan teknologi, menghasilkan karya inovatif yang berkualitas tinggi, sekaligus aplikatif dan tetap berkarakter kerakyatan, sehingga dapat diadopsi dan dimanfaatkan oleh masyarakat secara luas. Sumber daya manusia yang berkualitas tinggi didukung fasilitas yang baik menjadi keunggulan Fakultas Kedokteran Hewan UGM mewujudkan insan kesehatan hewan yang unggul di berbagai bidang seperti di bidang laboratorium diagnostik, entrepreneur, praktisi, satwa liar, ternak produktif, penyidikan zoonosis, penjaminan keamanan dan kehalalan produk hewan, hingga mengambil kebijakan dan keputusan di bidang kesehatan hewan. Demikianlah peran Fakultas Kedokteran Hewan UGM mewujudkan visinya yang mendasarkan profesi dokter hewan pada filosofi manusia meriga satwa sewaka, mensejahterakan manusia melalui kesehatan hewan.
Veterinarians are expected to take care of animal welfare, starting from providing food supply, quarantine, reproduction, treatment, until controlling the spread of contagious animal disease. Various tasks and expertises of veterinarian are learned in 12 departments of Faculty of Veterinary Medicine UGM. Department of Anatomy is one of the oldest departments in the Faculty of Veterinary Medicine UGM. The main lecturers in this department is the Principal of Animal Anatomy. Department of Anatomy has 2 laboratories, Macroanatomy and Microanatomy. Department of Biochemistry develops research related to genetic diversity. This department also focused on the development of the bird sexing based on DNA structures. Equipped with an advanced laboratory, the Department of Pharmacology is responsible for drug science, including pharmacotherapy and chemotherapy on animals, drug prescription, and research on drug effects on animals. This department also developed veterinary pharmacy, science in drug supply and drug management. Department of Physiology teach about the science of an animal's normal body function. The greatest research of Department of Physiology is the physiology response from various animals. Various development has been done in this laboratory, such as hormone testing and research on animal digestive nerves and development on stem cells. Department of Surgery and Radiology handles animal disease using advanced equipments in the veterinary medicine field, such as X-rays and USG. This department is facilitated with a special clinic for skill development and also as the community service unit. Department of Internal Medicine teaches about the clinical and laboratory examination techniques. The greatest research in the Department of Internal Medicine is the development of diagnosis and therapy for various veterinary internal medicine using advanced technology. Department of Veterinary Public Health plays a role in science of supplying animal source foods that are safe, healthy, intact, and halal, and also plays a role in the research of controlling infectious disease that can be naturally transmitted between animals and humans or zoonosis. Department of Microbiology develops science related to bacteria, virus, and fungus. This department plays a role in controlling important animal disease such as bird flu, rabies, anthrax, and SARS. Department of Parasitology develops research related to parasites such as worms and protozoa. This science plays an important role in controlling animal disease caused by parasites. The greatest research in the Department of Parasitology is vaccine production and molecular biological study on parasites. Department of Pathology has a laboratory that studies about cancer, diagnostic development on poultry and aquatic animals disease. Department of Clinical Pathology studies about animal disease diagnosis, drug side effects, and chemical materials. This department also develops research using animals based on proper science standards and animal welfare. Facilitated with an obstetric and gynecological laboratory, Department of Reproduction develops animal reproduction science, including reproductive disorders and reproduction technology. Various sustainable courses are held by this department, such as the inseminator and sterility courses. Besides advanced laboratories inside all of the departments, Faculty of Veterinary Medicine UGM is also supported by Animal Health Education and Training Unit or UP2KH. Until now, UP2KH managed several livestock such as cows, sheep, and horses. The facility in this unit is not only used by university students but also by veterinarians for the sustainable learning process. The learning process in the faculty always devoted by science and competencies to try to fulfill the society need. One of these is through research developed by the teaching staff, the discovery of the Regan chicken carcass testing kit that is fast and accurate. This testing kit has been patented and has widely used in the industrial scale, laboratory, and household scale. Faculty of Veterinary Medicine lecturers regularly write and publish books related to the veterinary medicine world that can be used for readers in the animal husbandry, biology, and statistics. The conducive learning atmosphere is proven effectively supports the university students to obtain various achievements, especially in the research field, arts, sports, and national and international organizations.
Dalam studi saya di fakultas ini, saya memiliki minat dalam bidang penelitian ataupun riset berbasis kedokteran hewan. Dan didukung penuh oleh Fakultas Kedokteran Hewan, saya mampu mengembangkan diri dan berprestasi selama belajar di sini. Melalui program kreativitas mahasiswa, atas bantuan dosen pendamping yang berkompeten, saya dan tim mampu lolos dan berhasil mengerjakan 12 proposal program kreativitas mahasiswa bidang penelitian. Mari terus berprestasi mahasiswa FKH UGM, manusia meriga satwa sewaka.
Halo, nama saya Ande Firmansyah, mahasiswa Fakultas Kedokteran Hewan Universitas Gadjah Mada. Selain aktif di organisasi lingkup fakultas, saya juga dipercaya menjadi pengurus inti dari organisasi mahasiswa kedokteran hewan global atau International Veterinary Student Association. Menurut saya, unit kegiatan mahasiswa yang ada di FKH UGM cukup banyak. Meskipun begitu, saya berterima kasih kepada FKH UGM karena telah membantu kami untuk menyalurkan minat, bakat, dan potensi diri kami. Besar harapan saya, dengan adanya bantuan ini, mahasiswa semakin aktif dan berprestasi. Manusia meriga satwa sewaka. Viva Veteriner Indonesia.
For the sake of developing the veterinary medicine science, the faculty established relations with national and international stakeholders. Faculty of Veterinary Medicine initiated the Veterinarian Medicine Association Indonesia through an MOU between the government and private sectors for research and community service. We also have collaborations with foreign FVM universities from Australia, Thailand, and Japan for research and education. Furthermore, we accept international students in Faculty of Veterinary Medicine, such as from Malaysia.
The reason I chose UGM is because it's well recognized and known for its academic excellence. My life in UGM, I would say, is tiring, but it's fun because I get to experience new things in class and also outside of class, such as my practical labs. The activities that I enjoy doing would have to be IVSF from the International Veterinary Student Forum, where we carry out activities such as Outbound, which allows us to not only bond with our seniors but also our lecturers.
I'm currently doing my 5th year in veterinary medicine in UGM. Why I picked UGM was because it's recognized in most countries, especially in Malaysia, and it's also in a city which can be very convenient. And the activities that I find interesting in UGM is the fact that the students get to teach the juniors a certain lab. For example, I'm currently an assistant in the lab called Veterinary Clinical Diagnostics. So what I learned from my seniors and from my lecturers, I actually carried on and teach to the juniors so that they also get the knowledge that I received from my seniors. So that's what I find interesting about UGM.
In order to pursue our dreams, we have done various community service activities. The Professor Suparwi Animal Hospital at Jalan Fauna No. 2 Karangmalang, Yogyakarta is one of them. The animal hospital is equipped with representative facilities and professional management. This animal hospital can become the reference of animal health care in Yogyakarta and its surroundings. Faculty of Veterinary Medicine UGM is committed to developing science and community service based on the veterinarian work principle Manusia Mriga Satwa Sewaka, which means we are to serve mankind through animal kingdom.
shining, shimmering, splendid. Tell me, princess, no, when did you last let your heart decide? I can open your eyes, take you wonder by wonder, over sideways and under, on magic carpet run, a whole new world, a new fantastic point of view. No one to tell us no, or where to go, or say we're only dreaming.
A whole new world, a dazzling place I never knew. But now from way up here, it's crystal clear that now I'm in a whole new world with you. Soaring, tumbling, freewheeling through an endless diamond sky. A whole new world.
Close your eyes.
100,000 things to see. I'm like a shooting star. I've come so far. I can't go back to where I used to be. Every turn a surprise. Every moment another. I'll twist it anywhere. There's time to spare.
to praying. Thank you very much. Class into 4 subsession, and during this parallel session, there is 2 moderator who will be moderate this session, and also we have the host and also co-host in this meeting room who will help if there is a problem during our meeting rooms. Now, let me introduce myself. My name is Hefi Hatmadetami as the first moderator. Dr. Andre, please.
Okay. Thank you for the introduction, Dr. Hefi. Hello, everyone. I'm Andre Fimasya, the second moderator of this parallel session in Class B, ICAVESS. Now, I'd like to read the rules of these parallel sessions. Okay, as you can see in the screen, so there will be 4 to 5 subsessions separate in this parallel session, which will be separated with Q&A session in each subsession. And in each subsession, there will be 3 presentation videos and 1 Q&A. And the video presentation will be around 8 minutes each. And participant may ask the presentation in the chat room with the following format: name of the presentation, name of the presenter, your question, and your email. We will do 1 round of a question, so 1 question for each presenter. But if we still have time of question, however, the remaining question will be sent to the presenter's email if time is not enough. And other technical issues may be asked in the chat room. We have Dr. Makruf Arif and Dr. Wari here for assisting. And during the session, please kindly mute your microphone unless you are asked to unmute them. To fill out the attendance form by the link provided in the chat box, and certificate will only be issued for those who attend all of the session in ICAVESS from the plenary and the parallel session. And now we will move on to the first subsession of this afternoon. Okay, we will have 3 presentations. The first one is a presentation on the biological variation, the index of individuality, and the reference change value for hematological and blood chemical analytes in ferrets or Mustela putorius furo by Yeremia Yobelano, followed by the proboscis monkey conservation in Suwi River, East Kalimantan, Indonesia by Triad Moko. Last but not least, a presentation about ex situ exploration of Cemani chicken in Balai Penelitian Ternak or Balitnak, Bogor, West Java by Tatan Kostaman. Okay, without further ado, co-host, you may play the video presentation. Okay, before the video presentation start, I would like to remind you to send your question while we play the video presentation in the chat box so that we can maximize our discussion time effectively. Thank you. Okay, you may proceed, co-host.
No voice ya, Dr. Ma'ruf.
Good afternoon, ladies and gentlemen. My name is Yeremia Yobelan, Faculty of Veterinary Medicine, University of Nusa Cendana, Kupang. In this 8-minute video, I will present my paper with the title, The Biological Variation Index of Individuality and Reference Change Value for Blood Chemical Analyte in Ferrets. So far, there are many published reference intervals, which are population-based interval in ferrets. However, there is no study about the biological variation of ferrets, whereas if we calculate the biological variation, we may find out that population-based interval is not always sensitive enough to detect pathological change for an individual. And the fact is the reference interval is apparently divided into the population and subject-based intervals. The population-based intervals is the common reference intervals that we usually use to analyze lab results like blood test result, and it represents cross-sectional data from a lot of individuals and consequently has wider intervals. Meanwhile, subject-based interval is the reference interval from a single healthy individual, which is specifically used for the same individual used to estimate the interval. And it represents the longitudinal data and usually has narrower intervals. So the question is, which one is better used or more accurate? Apparently, this question can be answered by calculating the biological variation, which are intra and inter-individual variation, and thus calculate the index of individuality. And another important value in this study is reference change value. It is the maximum, presumably normal difference between two consecutive measurements within the same individual. And together with reference intervals, the RCV is usually useful in making clinical decisions based on the lab results, especially for each analyte that has intra-individual variation lower than inter-individual variation. So, the objective of this study is to investigate the biological variation and the reference change value of hematological and biochemical analytes in ferrets. For the material and methods, this study was a retrospective study using annual hematological and blood chemical data of 13 ferrets in Bristol Zoo from 2010 to 2016. And the statistic method used in this study was restricted maximum likelihood to estimate the variance component, which are coefficient of intra-individual variation or CVI and the coefficient of inter-individual variation or CVG. And after calculating the CVI and CVG, the index of individuality was calculated using the simplified formula CVI divided by CVG, because we assume that CVA, coefficient of analytical variation, is significantly lower than CVI, as the blood samples were analyzed by automated analyzer in Bristol Zoo. And in this slide, showed the formula of the reference change value in this study. So there are three interpretations for index of individuality. If the index is lower than 0.6, it is better to use subject-based intervals, and the RCV is very useful for analyzing the difference of two consecutive measurements. And if the index is between 0.6 and 1.4, it is okay to use population-based intervals, but with caution, and the RCV may be useful enough. Last one, if the index is higher than 1.4, it is preferable to use population-based interval, and the RCV is not sensitive enough. So this is the calculation of biological variation and reference change value for routine hematological analytes in ferrets table. And then the next slide is the table for the blood chemical analytes. So the result In this study, no analytes has the index lower than 0.6, and the indexes of MCH, ALT, ALP, calcium, BU, and total protein, albumin, globulin are between 0.6 and 1.4. And while other analytes have the index higher than 1.4. And this figure shows the importance of understanding biological variation and reference change value. As you can see, the blue horizontal line shows the absolute range values for each ferret from 7 annual test result, and the black vertical lines, it represents the population-based interval for the 13 ferrets. And the total bilirubin in the right one represent the analyte with index higher than 1.4, and the population-based interval is preferable. The alkaline phosphatase or ALP here has the index 0.86, meaning that we can use population-based RI or interval with caution, because you could see in this figure for ferret number 1, for example, it's between around 10 to almost 20. It is very shorter, the absolute range value for the ferret number 1 compared to the population-based interval, which is around 5 to almost 45, around 45. So the RCV of the ALP, which is 82%, may help to detect significant or pathological change if the next test result is outside 10 to almost 20 intervals, but it is still inside the population-based reference interval. For example, if the previous result for the ferret number 1 is 15, then the next result is more than 82% or more than 27, such as 35, it is still inside the population-based interval, but it is wisely to think that the ferret may have problems. So the conclusion in this study are: first, MCH, calcium, BUN, ALT, ALP, total protein, albumin, and globulin in ferrets can be evaluated using population-based intervals, but this must be interpreted with caution. Then the RCVs calculated in this study may be useful to analyze the difference between two consecutive measurements of analytes mentioned before. Lastly, other analytes showed low individuality, making population-based intervals suitable for evaluating observed values for these analytes. And then here's the list of several references for this study, and that's all. Thank you.
Ladies and gentlemen, my name.
Okay, before we proceed to the next presenter, I would like to remind you that you can send your question through the chat box by a format the presenter name, your question, and your email. And friendly reminder for all the presenter whose video presentation is being played. Please turn on your video or camera. Thank you. So that we can pin you to the spotlight of this Zoom meeting. Okay, so for the next, Mr. Triatmoko, maybe you can turn on your camera so that we can pin you to the spotlight. Okay, there you go. Okay, we will continue to the second presentation.
Name is Tri Atmoko, and I am a senior researcher at the Research and Development Institute of Natural Resources Conservation Technology, the Ministry of Environment and Forestry. Today, I would like to present my paper with the title The Proboscis Monkey Conservation in Sui River, East Kalimantan, Indonesia. I work on this research with my colleague, Mr. Muklisi. Firstly, I will explain about the proboscis monkey. Proboscis monkey is endemic to Borneo that spread across 3 countries, Indonesia, Malaysia, and Brunei Darussalam. Proboscis monkey are protected species based on Indonesian government regulation. This animal is included in Appendix 1 CITES as an endangered species based on the IUCN red list. Most of the habitat are outside the conservation area. In other hand, their primary habitat destruction and change rapidly. The loss and destruction of this habitat have implication for the decline in the proboscis monkey population. This is a map of the distribution of proboscis monkey in Kalimantan. The riverine habitat in the Long Mahakam River is one of the important habitat for proboscis monkey. One of its tributaries is Kadang Kepala River and Konohan Sui wetland at the upstream as the location of this research. Sui River is part of the Konohan Sui wetland with its ecological and economic function. This area maintaining the hydrological balance with its essential of flood control. And this location is an area for the fulfillment of local people livelihood. This location become the habitat of protected endangered species, proboscis monkey and Siamensis crocodile. This study aimed to determine the distribution, habitat, and potential threats to proboscis monkey. This information is essential as a basis for conservation effort. The research was conducted in July 2017 in Sui River, East Sakutai Regency, East Kalimantan. To find out the distribution of proboscis monkey using a bird survey method, vegetation analysis was carried out to determine the structure and composition of plant in the habitat. Further identification of plant species was done at the Herbarium Wana research. The result that is so there were 12 groups of proboscis monkey that spread along the Sui River. This study has not been able to count the existing proboscis monkey population. In this proboscis monkey habitat, there are at least 60 species of plants, 30 of which were found in the observation plot, and the description of 55 plants have been published in this book and can be accessed freely at the following link. The vegetation on the habitat The tree level is dominated by Malothus sumatrana, Lagerstormia speciosa, and Glutarenus. And the Malothus sumatrana and Barringtonia acutangula dominate in vegetation at pole and sapling level. Two species of plant have potential to be invasive species, namely Mimosa pigra and Salvinia molesta. There are at least 12 species of Proboscis monkey food plant at the research location. That is Lychea, Glucidion, Cariatia, Gardenia, Mangifera, Cludia, Garcinia, Barringtonia, and Malothus. Some of the threats to the Proboscis monkey in the Sui River are illegal hunting, conversion of habitat to oil palm plantation, forest fire, and occupancy of invasive species. The conclusion of this study, the Proboscis monkey habitat comprise more than 60 species of plant that still support to Proboscis monkey. The habitat destruction caused by fire need to be rehabilitated using the native species, and be aware of the presence of invasive species, and habitat loss and destruction need to be prevented by collaboration management as an essential ecosystem area. Thank you very much.
Okay, next. Okay, we already have Mr. Tatan here. Okay, before we play our presentation, friendly reminder again to send your question through the chat box so we can save some times. Okay. Yeah, you can continue.
Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, first we want to thank you all for your presence in this seminar and today's presentation. Before we begin, let us introduce ourselves. My name is Tatan Kostaman. I am from Balitnak, Bogor, West Java. The title of this presentation is Ex situ Exploration of Cemani Chicken in Balai Penelitian Ternak, Balitnak, Bogor, West Java. So first, Cemani chicken is one of the is usually used for religious or traditional ceremony. However, in their natural habitat in situ, the population of Cemani chicken is not known. To maintain the Cemani chicken population, step or program that can be taken are by conducting a germplasm management program. The conservation of conserving the genetic strain of endangered species and those of commercial value. Finally, the purpose of writing a paper is to explore Cemani chicken that is kept ex situ in Balitnak in terms of production, weight, egg quality, and growth of Cemani chicken, 4 days of chicken until the egg of 20 weeks, so that this paper can be useful to supplement information on the development
of Cemani chicken outside their habitat. The research was conducted at Java. Team was carried out with a male to female ratio of 1:5. Provision of commercial feed as much as 100 grams per head per day. A total of 20 hens and 4 roosters were used. Second, eggs were collected manually between 9 to 11 hours. Then, freeze on the tray. Sorted eggs, which are clean and of good quality, are collected for hatching purposes. Third, eggs were placed in an automatic portable incubator at temperature of 27.8 degrees Celsius and 70% relative humidity. On the 18th day of incubation, candling was performed. Furthermore, fertile eggs are transferred from the incubator tray to the hatching tray on the same machine. Furthermore, the parameters to be viewed are hen production, first egg weight, egg quality, and growth of Cemani chicken. Raw data was entered into the Excel program. Calculation of mean and standard deviation was performed using the help of the SPSS version 25 program, and the results were tested descriptively, which were in table and graph. Hen-day production percent of Cemani chicken during the 6 months increased from 14.96 to 48.97%, and is almost the same as black Kedu chicken. That is, the Cemani chicken was able to be developed in the Balitnak area ex situ. First egg weight, Cemani chicken was 31.31 gram and relatively higher than the weight of black Kedu chicken, egg of 28.64 gram. The higher weight of the first egg in the study was probably by the quality and amount of feed given. The egg quality is something that is seen and observed to compare whether or not the egg is good or bad. The high quality of Cemani chicken is higher than the black Kedu chicken. The average weight of Cemani chicken egg yolk is higher than that of Dayak local chicken, 11.7 gram. The average albumin weight of Cemani chicken is also higher than that of the Dayak local chicken, 14.78 gram. With sea ground, the weight of Cemani chicken has a lower head weight when compared to Abdurrahman research showed that the hatching weight was 28 to 32 gram. At 5 months, the weight is higher. According to Abdurrahman, the weight of Cemani chicken at the age of 5 months, namely 1,400, 1,500 gram male, and 1,200, 1,300 gram female. In conclusion, everything that genetic resource exploration activity are still need to maintain the diversity of extant animals. The research can be provide information about the structure and quality of Cemani chicken eggs, which can be used as a basic for the development and refining program of Cemani chicken outside their habitat ex situ. Thank you very much for your great attention, participation.
Okay, so thank you for all the video presentation. That was all an insightful presentation from the first three presentator. So now we will move on to the Q&A session of this subsession. So we will start with the first round of question, which one question for each presentator. Let's see on the chat. Okay. Okay, I think questions are coming up. So I'd like to open this first round of Q&A session with a question for the first presentator. On your camera so that we can be
for the times you give it to me, and then I will start the second subsession for this Class B parallel. Before we start the presentation, I will invite, or we
Beach in Gunung Kidul, Yogyakarta. the picture H, it has black brittle star with reddish tube feet and 2 tentacle scales. The picture I, Opiocomas colopendrina, has brown discolor and brown arm with various pattern with brown and uniform tube feet. And the last is Holoturoidea classis. We only found Actinopica echinetes, the picture G. So it has characteristic medium size. It has various color body from light brown into orange, something like that. It's rarely found in Krakal Beach, Gunung Kidul, Yogyakarta. So last, the conclusion of the research that we found. There we found 10 species of echinoderms, which are divided into 3 classes. The Echinoidea classis consists of 7 species. There are Stomopneustes variolaris, Echinometra matei, Trypneustes pendricosus, Trypneustes gratila, Echinotrix calamaris, Diadema setosum, and Diadema antillarum. The Ophiuroidea classis consists of 2 species, Opiocomas erinaceus and Opiocomas colopendrina. The Holoturoidea consists of 1 species, it's Actinopica echinetes. So this is the end of my presentation. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you for the presentation from the first presenters. Before we going to the second presentation, I will welcome to Dr. Heri Wijayanto as the second presenter for this session. Maybe Dr. Heri can turn it on the camera so we can put it together in here. And then I also ask or invite all of the participants in this class if there is any question. There is interesting research here. We know that in the Krakal Beach we have so many echinodermata, and the name is very beautiful. Don't use as your son or daughter's names, otherwise your son or daughter's names will be in the names of echinodermata. Okay, we start to our second presentations. Please, the co-host, the presentation from Dr. Heri Wijayanto, the locomotion study of the Bekantan and at Koryak Island.
Good afternoon, everybody. My name is Heri from Faculty of Veterinary Medicine, Gajah Mada University. And today I would like to present small observations in Kantan or Natural South Kalimantan in collaboration Labung Mangkurat University and Natural Resources Conservation Bureau of South Kalimantan. And we know that according to the CITES, Bekantan conservation status is endangered. And in South Kalimantan, we can find the Bekantan who still freely ranging in their natural habitat at Bakut Island and Curiak Island. And our observations was done in Bekantan Research Center, Curiak Island, Peritukuala, South Kalimantan. Bekantan or Nasalis larvatus is distributed in the whole Kalimantan, and there are 2 species of Bekantan, Nasalis larvatus, who distributed in the whole Kalimantan, and Nasalis larvatus orientalis, who distributed in East Kalimantan. And we know that Bekantan have a special locomotion organ because they have partially webbed fingers and toes, and it believed very important for swimming. And beside that, Bekantan also have a long and big nose with furnished with pulp and shark nose, we perhaps is very important in their water activities. This figure shows us how the web fringers exist in Bekantan. Morphological study and morphometric study have been done on 2019, and from that study we found that between the length of femur and the body length of Bekantan have very strong correlation. And also we found that the brachial index of Bekantan is 90, means the locomotion is supposed to be a hind limb dominant. But on the field, we can find that Bekantan not only can move bipedally, but they also move easily quadrupedally, hanging or semi-brachiation, and also jump. Here is the figure that shows us the daily activities of Bekantan at Curiak Island, and the photographs were kindly provided by Sahabat Bekantan Indonesia. And like other groups monkey, Bekantan also led by an alpha male. And we look at this picture, this is the alpha male of the groups. And locomotions, they move quadrupedally, but on the other time, they also can move bipedally. So both of the movement can be done by Bekantan easily. However, the quadrupedally movements look like more often compared to the bipedally movement. And Bekantan also spend most of their time on the trees, and they also good climber on the trees. And this is the serial picture that show us how the dominancy of the hind limb in the movement. When they need extra energy to move, like this picture, they start to jump using the power of hind limb, and they also will be landed with the hind limb. So this is show us how the hind limbs more dominant. And Bekantan also a very good swimmer on the water, even diving. And it looks like the long and big nose is the result of long evolutions to support their activities in the water. However, nowadays, the water activities is very rare, and perhaps they avoid the water predator, especially for the infant and juvenile. And it looks like their ability in locomotions is the result of long evolutions. And as we know that the evolutions is influenced by genetic and environmental pressure, and it looks like the environmental pressures is push the Bekantan to develop the certain organ for their survival. And this theory is very appropriate with the evolution theory by Jean-Baptiste Lamarck, where the the theory is stated about the use and disuse organ. The use organ will be developed and the disuse organ will be reduced or atrophy. Also, their nose is thought to be closely related with water activities because the nose is varnished by a kind of valve. Thank you very much for your attention. And also thank you for the Dean of Faculty of Veterinary Medicine, Gadjah Mada University, Biology Study Program, Faculty of Science, Lambung Mangkurat University, Sahabat Bekantan Indonesia, Natural Resources Conservation Bureau of South Kalimantan, and also all of my friends in Anatomy Department, Faculty of Veterinary Medicine, Gadjah Mada University. Thank you.
Thank you for the second presentation. So before we enter to the third presenters in this second subsession, I'll remind about this presentation, two presentations before. It's very interesting about echinodermata and also the Bekantan nasalis larvatus. So please, so we can develop our nice discussion later. So for the co-host, please, for the third presentations.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everybody. I would like to thank for opportunity it's given to me to present my research with title Micromorphology of the Digestive Tract of Monitor Timorensis, Varanus timorensis. We are from Department of Biology Education, University of Muhammadiyah Kupang, Indonesia. Introduction. Timor monitor lizard is one of the species from the Varanidae family, is Varanus timorensis. The distribution of this animal in Indonesia is in Timor Island and on the surrounding small island like Rote Island, Small Island, and Sabo Island. Special feature on morphology. Has a slightly cylindrical tail, nostrils ridge, rounded nostril that located close to snout than to eyes. Dorsal scales have a greenish-brown colors with a yellow round pattern. Population this animal with decreasing cause of hunting for commercialization. The scientific information has not been widely reported. Therefore, this research aims to determine the micromorphology of the digestive tract of the monitor timorensis, Varanus timorensis. Material and method. The sample we get from Kevanan forest from Timor Island. Location. This research was conducted in the laboratory of the Biology Education, University of Muhammadiyah Kupang for the tissue perfusion process and histology preparation and hematogenation staining in the laboratory of Siruang Hospital Kupang. Methods. The first, tissue perfusion, histology preparation, and hematogenation staining. Material and method. Procedure. The first, Varanus timorensis will be anesthesiated. Next, perfusive process. Next, histological preparation. Next, hematogenation staining. The last point, observation under light microscope. Result and discussion. Microscopic observation of esophagus. From the tile we can see in table 1. We can see from the esophageal wall layer, we can found the fourth layer is mucosa, submucosa, tunica muscularis, and serosa. To be clearly, we can see that figure 1, histology structure of monitor timorensis esophagus. Microscopic observation of the esophagus. The mucosal layer of the esophagus is the deepest layer into the lumen that consists of epithelial mucosa, lamina propria mucosa, and tunica muscularis mucosa. The epithelial mucosa tissue composite of multi-layer flat cell with the oval nuclei shape. The structure of esophagus tissue of monitor timorensis is similar to other familiar reptiles, such as a rainbow snake. The lamina propria mucosa composite of connective tissue that separate epithelial mucosa and muscularis mucosa. The lamina muscularis mucosa composite of the very thin smooth muscle. The submucosal layer that distributes in all parts of esophagus look narrow and composite of the loose connective tissue. Microscopic observation of stomach. We can see in table 2 histological structure of the stomach wall layer of monitor timorensis. We can find the four layer. The first mucosa layer, submucosa, tunica muscularis, and serosa. To be clearly, we can see the figure 2 histology of structure of monitor timorensis stomach. Microscopic observation of stomach. The stomach of monitor timorensis is an extension from anterior intestine and have monogastric type. The microscopic structure of the stomach consisted of mucosa layer, gland, submucosa, tunica muscularis, and serosa. The mucosa layer of the stomach consists of epithelial mucosa cell, lamina propria mucosa, and muscularis mucosa. The muscularis mucosa composite of circular smooth muscle. 3. Microscopic observation of intestine. We can see in table 3 the histology of structure of the intestine wall layer of monitor timorensis. To be clearly, we can see the figure 3, the histology structure of monitor timorensis intestine tissue. The intestine of monitor timorensis consists of small intestine and large intestine with no cecum as a border that's similar to water monitor. The presence of cecum in a reptile correlates to their eating habit. The mucosa layer of monitor timorensis intestine consists of epithelial mucosa, lamina propria mucosa, and tunica muscularis mucosa. The epithelial mucosa of the intestine consists of monolayer cylindrous epithelial cell and goblet cell are in between the epithelial cell. The villi length of monitor timorensis is changed gradually in accordance. The tissue structure of the lamina propria in all parts of intestine is similar. The lamina muscularis mucosa are thin that composite of circular smooth muscle. The tunica mucosa of monitor timorensis intestine is similar to that of the water monitor. The structure of submucosal layer in the intestine of monitor timorensis is similar to rainbow water snake and water monitor. Conclusion: the digestive tract wall of monitor timorensis consists of some layer, mucosa, gland, submucosa, tunica muscularis, and serosa. Except in esophagus, no gland was found there. Thank you for your attention.
Thank you very much for the presenter presentation. Now we are in the time for question and answer Q&A, and then so there is so maybe I ask once again. Okay, please, Dr. Harry, if you can first, please turn it on your camera. So now we will going to the question. Yeah, I saw there is some question for the presenters. I will share my screens for that one. So the presenters can see clearly and the question. For the first question is for Ms. Salma Dewi Pratita. This question comes from Mr. Mahfud, and he asks, how about the diversity level? For example, number of individu per species of Echinodermata in the beets by using the formulation to calculate the diversity. So because in your presentation, he say you just reported about kind of species. Please, for you to answer the question.
Okay. Thank you, Mr. Mahfud, for the question. We don't diversity level, so we only take some species and identifying the species, but we don't calculate the number of individual per species. But maybe in the next research, we can continue and complete the data by calculating number of individual per species. Oh, is my voice clearly?
Up and down, you can repeat.
Oh, sorry.
It's
sorry.
okay, no problem.
Yes. But now it's
it's clear. It's clear now. You can repeat.
Thank you. About our research, we unfortunately we don't calculate the number of individu per species, but maybe in the next research we will do that with the richness or something about biodiversity. So maybe we can complete the data in the next research.
Okay. Mr. Mahfud, I think maybe for because of restricted time, for more longer detail, you can contact Ms. Salma for more discussion. So thank you very much for your answer, Ms. Salma. And then I'm going to our second presenters, Dr. Heri Wijayanto. There is two questions. So the first is there from Yeni, Miss Yeni? Is there any evidence that the ancient proboscis monkey didn't originate from variation as stated by Darwin? The second one is there a correlation between body and leg length in ape primates to its natural surroundings, the large nose function as a consequence of the proboscis monkey's swimming proficiency? Okay, Dr. Heri, please, the time for you.
Okay, thank you very much. Can you hear my voice?
Yeah, clearly. Thank you.
Yeah, thank you. The first questions from Mrs. Jenny. Actually, there are three theories of evolutions, but all of the evolutions it look like depend on the genetic and environment pressure. So I think the big and long nose in Bekantan is not only variation, but this is part of the natural selections. As we know that the bigger male nose will be more dominant compared to the smaller. So the alpha male, if you see, they must have the biggest size of the nose in the group. And you know that by this, what can I say? They need, by this needed, they will push naturally how to select the alpha male just by the nose size in bekantan. Because you know that the stronger alpha male will be the leader, but they're not only stronger, but also must have the big nose in bekantan. So that's why I hypothesize that this big nose is supporting the water activities in bekantan. And as I said, that the most suitable theory for in bekantan, I think Jean Baptiste Lamarck by use and disuse organ statement. I think that was one from for Mrs. Yeni, and the second one.
The correlation between body and leg length
Okay.
in ape primates.
We know that the size of any organ of animals will be developed in very, very long long period. And you know that evolution is very long adaptation. And is there correlation between body and leg length in ape primates? To its natural surrounding. If we look at orangutan, for example, as the ape, you know that orangutan not fully brachiation in movement because they have heavy weight body. By these conditions, they will develop another capabilities to move not only by brachiation, but they also climbing and quadrupedally walking on the ground. That's for great apes. And as we know that because any part of the body have proportions, their own proportions in anatomically. So if you collect data from the leg length and the body length, they must have close correlations. But you must note that the data must come from the adult, not from infant juvenile, because the infant and juvenile is not reached the fully development yet. And the large nose function as a consequence of the proboscis monkey swimming, yes, I have what can I say? It looks like they need the big nose very much in the water activities because there is the structure valve-like in the nose, and this structure, I believe, is very important when they swimming or diving in the water. And this is I think the only monkey who have good activities in water. But as I said now, it's very rare because perhaps predator. But I still trying to get the documentations on bekantan water activities this year. Thank you.
Okay. Thank you very much for the answer, Dr. Harry, and also Miss Salma. Miss Salma, yeah, correct, Miss Salma. Okay, and then we going to the third presenters is more interesting, yeah. Please for Mr. Germanus, there is the last question for this session. Can the summary is how you perfuse your lizard from Miss Golda Saragi. The question is, please. Mr. Germanus, are you there? Hello. Oh, I think maybe there is a connection problem to him. Wait. Dr. Ma'ruf, can you check? I cannot see him anymore in our list of participants.
Sebentar saya.
Okay, maybe because of our restricted time, maybe we can go into the third subsession, and the question from Ms. Golda Saragi, we will ask Mr. Germanus privately, and we'll send to you the answers. Please, we give a virtual applause to all our presenters this evening. A very interesting presentation from Echinodermata to the Bekantan and lizards. Thank you very much. And then I give the time for Dr. Andre to moderate the next sessions. Thank you very much.
Okay, thank you, Dr. Heavy. I think it's getting warmer here for the discussion. I saw so many questions coming up, and I hope we will have more questions for the last two subsessions this afternoon. Okay, moving on to the third subsession of this parallel session. I would like to ask three presentators to turn on their camera. So I would like to ask Mr. Mahfud, Ms. Okti Herawati, and Dian Bekti Hadi Masito, so that we can pin you to the spotlight. From Mahfud, all right. Here. Okti and Dian Bekti. Wait, it's here, right? Okay, we are all here. So good afternoon, everyone. I hope we will have a good discussion for this third subsession. And without further ado, for the first presentation, we will have a presentation about histochemical study of the esophagus and stomach of water monitor lizard, or Varanus salvator, from Mr. Mahfud. And the second presentation will be from Ms. Okti Herawati in the presentation of antibiotic resistance from different locality of wild captured Coura amboinensis. And the third presentation from Dian Bekti Hadi Masito about the anatomy of Sunda porcupine, or Hystrix javanica adrenal gland. Okay. Okay, co-host, you may play the video presentation.
Hello, Assalamualaikum warahmatullahi wabarakatuh. My name is Mahfud. I will present the result of my research entitled Histochemical Study of the Esophagus and Stomach of Water Monitor Lizard, or Varanus salvator, or reptile Feridae. Varanus salvator, or as known as water monitor lizard, or as known as Asian water monitor, is one of member of Varanidae family. This species is distributed from South Asia to Southeast Asia, including Indonesia. The world's second largest lizard, after Komodo dragon, or known as Varanus comodensis. Because of its large spreading and its high status appendix 2, the hunting activity for commercialization is still frequently down. More than 1 million taken in natural. If this continues, it will cause a decline in this animal population to near rare. Therefore, we need an alternative solution, namely conservation efforts, so that this animal is not taken continuously. Some information of this animal has been reported. Generally, the research is limited in the study of its ecology, body morphology, and commercial harvesting. The information about the digestive tract of Varanus sarcopter has been reported before, namely about macroanatomy digestive tract, histology of the intestine, and immunohistochemistry of endocrine cells. Research method. The animals were anesthetized, exsanguinated, and fixed using paraformaldehyde 4% by perfusion method. The obtained tissue samples were then prepared for histological evaluation, paraffin method, with section thickness 3 to 4 micrometers. Then standard using hematoxylin eosin and casontricum, Elysian blue, and periodic adhesive. Microscope observation, we using light microscope with microphotography apparatus. Result and description. Morphology of esophagus, macroscopically, the tissue that connects directly to the lumen is mucosa tissue that composed of stratified columnar cylindrical epithelium. This stratified columnar cylindrical epithelium, which is its nucleus, located in the basal of cytoplasm. The esophagus mucus is also supported by lamina propria. This lamina propria that dominated by connective tissue and supported by longitudinal muscle. Morphology of gastric or stomach. Macroscopically, the stomachs of Varanus sarcopter is different animals that have Varanus sarcopter have straight shape of stomach, straight shape of stomach like the esophagus, and have bigger size than any other digestive organs. The result of microscopic observation show that the stomach wall of Varanus sarcopter composed of 3 layers. Tunica mucosa, glandular, and tunica muscularis, as similar as vertebrae. The tunica mucosa composed of lamina epithelia, consisting of stratified columnar cylindrical epithelium. The gland cell was found in the tunica glandular. This tunica glandular, the gland cell gives an overview about the type of the end of gland tubular echinor or tubular echinor. The intensity of the mucous substance in the stomach of Fahrenheit sulfur, the assembly at pH 2.5 and periodic adhesive staining result in the stomach of Fahrenheit sulfur show that the epithelial mucosa and lumen of the gastric gland secrete high concentration of natural mucopolysaccharides and do not secrete acid mucopolysaccharide. Inclusion in general, the tissue structure of the esophagus and stomach of Fahrenheit sulfur is similar to the common vertebrae. The esophagus of Fahrenheit sulfur has no glands and no keratinization process. The gland area, like cardia, fundus, and pylorus, also was not found in the stomach of Fahrenheit sulfur. The epithelial mucosa and lumen of gastric gland secrete high concentration of the natural mucopolysaccharide and does not secrete acid mucopolysaccharide. This is presumably because of Fahrenheit sulfur is a carnivore that swallows food directly without chewing it first. Acknowledgement 1. The study was funded by the Ministry of Research and Technology of Indonesia through the program of Novice Lecture Research Grant 2007. 2. Mrs. Roro Sri Catur Setiawaningsi, MSc, who was donated 2 male Fahrenheit sulfur pivotatus as samples in this research. There are other studies. It has been published in the international journal. Alhamdulillah, thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you for the presentation, Mr. Mahfud. We will continue to the second presentation. I would like to remind you again to send your question to our chat box using the format the presenter name, your question, and your email. Thank you. Okay, we will move on to the next presenter.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Thank you for this opportunity. Today, I would like to present our study about antibiotic resistance from different locality of wild capture Cowra amboinensis. We know that Indonesia has a land area covering 1.3% of total area from the world, and Indonesia also has 511 species of reptile, and nowadays reptiles are widely used as a pet, especially Cowra amboinensis. The increasing popularity of turtle to become pet lead increased case of zoonotic disease. The disease that often occurs is bacterial infection. Therapeutic without susceptibility testing caused the turtle given broad-spectrum antibiotic. Disease can make risks to antibiotic resistance. Antibiotic resistance is currently global concern. It's due to treatment value, decreased drug of choice. If the resistant antibiotic continue to widespread and drug of choice will be difficult to find, which can make increase the mortality of Cowra amboinensis. So our study was conducted to determine profile of resistant antibiotic in Cowra amboinensis. The result expected to provide scientific information regarding drug of choice that can be used to treatment. so we can play a role prevention and control of antibiotic resistance to conserve Cowra amboinensis. This is the material that we use in our study. We use 20 Cowra amboinensis from different locality, Sumatra, Kalimantan, Sumbawa, and Sulawesi. We use 8 different antibiotic tips, and we also use different medium to cultivation, identification, biochemical reaction, and also testing antibiotic resistance. This is the method that we use in our study. All methods will be confirmed by ethical clearance committee, and this is the number of ethical clearance letter. From 20 local swab, we cultivation in brain heart infusion, and then we transfer to Mekong agar to identify the colony bacteria, and then we transferring to biochemical reaction to identify the bacteria isolate, and then isolate bacteria testing antibiotic resistant by Gibbs-Bauer method, and then we determine the antibiotic resistance by diameter of inhibition zone, and then the result will be analyzed by descriptively analyzed. Based on our research, it can be found that isolated bacteria from several Cowra amboinensis has various antibiotic resistance. In general, antibiotic resistance to chloramphenicol reach 5%, and this is the lowest one. And then ampicillin 35%, oxytetracycline 15%, streptomycin 50%, ciprofloxacin 40%, amoxicillin 30%, gentamicin 25%, and colistin, the highest one, 75%. When we compare to the previous study, we know that oxytetracycline, ampicillin, and colistin that have different results from previous study, but ampicillin and oxytetracycline, when we compare the previous study, has presented lower than before. This is a graph about profile resistant antibiotic from different locality. We can see that colistin is the highest resistant in Cowra amboinensis from Sumatra, Sulawesi, and Sumbawa, but different with Cowra amboinensis from Kalimantan. Cowra amboinensis from Kalimantan have highest resistant antibiotic to ampicillin, reach 60%. According the difference of percentage of antibiotic resistance is influenced by environment. Antibiotic pollution in environment is effect of overuse of antibiotic in agriculture, medicine, or veterinary medicine. So different pollution of antibiotic resistant in environments can conduct different percentage of antibiotic resistance in Cowra amboinensis. Besides it, intrinsic factor from bacteria also influence the antibiotic resistance in Cowra amboinensis. we can conclude our study that profile antibiotic resistance in bacteria isolated from various locality of Cowra amboinensis were different, and the highest antibiotic resistance is colistin, 75%, and the lowest antibiotic resistance is chloramphenicol, 5%. So we can advise that chloramphenicol can be used the best drug of choice to treatment bacterial infection in Cowra amboinensis. Okay, this is a reference that we use in our study. Thank you for your attention. If you have any question, you can ask me directly. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Thank you for the presentation, Ms. Okti Herawati. Now we will move on to the last presentation of this third subsession. Co-host, you may play the video presentation.
Good afternoon, my name is Dian Bakti Hadiwasito and I will present our research about the anatomy of Sunda porcupine adrenal gland. Our research team are Dian Bakti Hadiwasito, Irma Padita, and Teguh Budi Pitojo. Sunda porcupine or Histix javanica is a species of rodent in the family Hystricidae. It's endemic in Indonesia. Due to the popularity of hunting and consumption, Indonesia Ministry of Environment and Forestry has listed Sunda porcupine as a protected animal since 2018 and depend on International Union for Conservation of Nature. Conservation status of Sunda porcupine is least concern. Our present research are digestive system, reproductive system, and integument system in Sunda porcupine. The research to determine the structure of Sunda porcupine adrenal glands. In this research, we use 9 Sunda porcupines from Tawangmangu subdistrict. After that, tissue processing with paraffin method and hematoxylin eosin staining. This is the result of adrenal gland Sunda porcupine. Topography of adrenal gland Sunda porcupine. The adrenal glands are located in abdominal cavity. The size are small, a pair, and located in anterior kidney. The left adrenal gland in Sunda porcupine larger than the right. In mice, the adrenal glands are ovoid and small in small size. In cattle, the right adrenal gland is triangular or heart-shaped, while the left adrenal gland is sometimes sarcoma-shaped with the length of the two glands usually varying from 5 to 8 centimeters with an average width 3 to 6 centimeters. This is the eosin hematoxylin staining. In the hematoxylin eosin staining, so that the adrenal gland have two parts, medulla and the cortex. The cortex consists of 3 zones. There are zona glomerulosa, fasciculata, and reticularis. The glomerulosa is the outer zone of the adrenal cortex. The cells in this zone are oval-shaped, separated by thin connective tissue from the fibrous capsule, and the zona fasciculata is composed of column-shaped cells that are oriented radially toward the medulla. The reticular zone is located directly adjacent to the medulla. Small cells form the irregular pattern and are separated by capillaries and connective tissue. The adrenal glands were in the abdominal cavity, small, open, and located anterior to the kidney. The adrenal glands have two parts, medulla and cortex. The cortex consists of three zones, glomerulosa, fasciculata, and reticularis. Thank you.
Okay, that was an eye-opening presentation from the presenter. We will now proceed to the Q&A session. So we already have some questions. So okay, I will share the screen. All right. Okay. I hope you can see my screen now. So first question for Mr. Mahfud. Is there any correlation between the animal's diet and the organ's morphology, and which staining method is the most difficult to be done, and why? Thank you. This question is from Ms. Saragih Golda.
Thank you for question. I think yes, there are a difference because animals, all animals, it adapts to type of food, and then I think the most difficult stain are Alcine blue and predecessive because can I ask for English language?
Yeah.
Yeah. Why difficult?
Miss Golda. Miss Golda, you may unmute yourself.
Hello.
Hello. Sorry, can you please repeat the question again?
Can I explain to English language in bahasa Indonesia?
Oh ya, boleh.
Jadi pertanyaan tadi, apakah ada hubungan antara pola makan atau jenis makan terhadap morfologi organ? Jelas ada perbedaannya karena yang pertama berdasarkan jenis makanannya. Kita tahu bahwa biawak itu kan atau rata-rata reptil pemakan daging atau karnivora pasti berbeda dengan hewan yang misalnya pemakan tumbuhan atau herbivora. Jelas tadi dari bentuk giginya ya sampai ke bentuk lambungnya. Herbivora rata-rata misalnya pemangsa biak atau ruminansia memiliki 4 lambung. Kemudian rata-rata yang omnivora juga itu ada curvatum major, ada curvatum minor, sedangkan pada biawak itu tidak ada curvatum minor dan curvatum majornya. Jadi bentuk lurus saja. Jadi rata-rata saluran pencernaannya lurus, tidak ada curvatum major maupun curvatum minor. Tetapi secara histologi atau mikroanatomi susunannya hampir sama. Susunan secara histologi hampir sama, ada lapisan mukus, kemudian lapisan epitel, kemudian ada lapisan dan juganya itu kelenjarnya juga ada otot. Kemudian apa yang paling sulit, pewarnaan yang paling sulit itu ya pewarnaan karbohidrat ya atau substansi mukus, yaitu pewarnaan Alcine blue dan periodic adhesive. Itu yang menurut saya paling susah. Kemudian apakah saya boleh menjawab pertanyaan tadi untuk Germanus? Kebetulan kami tim peneliti, apakah saya bisa menjawabnya?
Yes, you may. The question about perfusion, right?
Ya.
Yeah, okay.
Oke, yang pertanyaan untuk Jerman tadi bertanya metode perfusi ya. Ya, jadi kebetulan biawak yang saya teliti dengan yang Jermanus itu sama, ya hampir sama. Bedanya kami, kalau yang saya biawak itu menggunakan kombinasi ketamin dan silasin. Nah, kebetulan yang Jermanus ini, biawak timur ini karena penelitian di Kupang, kami susah mendapatkan ketamin dan silasin, jadi kami menggunakan namanya kloroform waktu itu. Ya, kloroform, kemudian kami untuk anestesinya menggunakan kloroform, dia kami suntik melalui intraperitoneal otot perut waktu itu. Kemudian karena hewannya agak, jadi biawak timur ini ukurannya kecil, nah, jadi kami perfusi eksaminasinya, kemudian perfusinya itu melalui jantung ya. Jadi tidak melalui pembuluh darah di tubuh, tapi melalui atrium kanan jantung. Jadi kami langsung menusukkan kanul di atrium jantungnya. Jadi proses eksaminasi sampai perfusinya itu melalui jantung. Mungkin itu tambahan dari saya. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh.
Okay, thank you for the answer, Mr. Yeah, I think it's quite complete, right? And thank you, Golda, for the two questions from two subsessions. We will move on to the next question for Ms. Okti Herawati. We have two questions here. Let me share the screen again. All right. Okay. Sorry. Okay. I hope you can see the screen, Miss. So for question number one, do you need KLHK permission for this research? And have you identified what bacteria are found in the cloaca? And there's a follow-up question here. Why is there antibiotic resistance in wild animals while therapy in wild animals is not widely used? Okay, Ms. Okti, you may answer the questions.
Okay, thank you for this question. First, I just have ethical clearance only. I not have KLHK permission. And in this study, I just isolated one bacteria, Escherichia coli, and I'm not identifying all the bacteria in the cloaca. So in this study presented of resistant antibiotic of Escherichia coli. And then the second question, because I want to know the antibiotic contaminant in environment, so we identify resistant antibiotic in turtle, because the turtle not use antibiotic in therapy. So we want to know the environment enhance antibiotic resistance in the turtle. Maybe it can be answered a lot of this question. Thank you.
Okay, thank you, Ms. Okti Herawati, for the answer. And maybe it's exotic animals. Okay, for the last question for the last presenter. Wait. Okay. This is for Ms. Dia. So, is there any differences between Sunda porcupine's adrenal gland histology and other rodents? Ms. Dia, you can unmute.
Thank you for the question.
Yeah.
The difference between Sunda porcupine adrenal and the other rodents, the basic similar, because Sunda porcupine is a rodent too. The difference is just the size. With mice, the Sunda porcupine adrenal glands is larger than the mice, and with rats, similar size, but depend on the size of Sunda porcupine. If the Sunda porcupine have the large size, the adrenal gland is larger than the rats.
Okay, so it's basically about the size difference between those rodents, right? Okay.
Yeah.
Okay, Ms. Dia, thank you for the answer, and thank you for all of the three presentators in this subsession. It was a fun discussion. Well, we still have one more subsession, which will be moderated by my co-moderator, Dr. Heavy again. So, Dr. Heavy, you may take over.
Thank you very much for the last subsession 3 presenters, and then I will going to our last subsession in this class B parallel. So we only have two presenters, yeah, and hopefully it's still going interesting to others in here. So I will invite, we will invite Anastasia Stefani for the presentation entitled Correlation Between Foot Circumference and Body Length, Shoulder Height, and Pelvic Weight in Sumatran Elephants, Elephas maximus sumatranus. Please turn it on your camera. And also, thank you very much. And also for Wahyu, Miss Title Dynamics of Diversity Decapods in the Intertidal Zone at Porok Beach, Gunung Kidul, Special Region of Yogyakarta, Indonesia. So, thank you for coming, and then please turn it on the camera for Miss Wahyu Febriani also. Before we start our presentation, happens in here form in the link which is present on the chat box, please, because we have 34 participants in here, but there is our record is still only 25 from this group. So maybe in here is if there is somebody haven't yet filled, then the hello, Anastasia and Wahyu, Miss Wahyu and Miss Anastasia. So both of you ready, yeah? The girl in the last presentation session. Okay. Now for Dr. Ma'ruf, please present the presentation 1 from Miss Anastasia.
My name is Anastasia. I am the representative for ICAVESS about the study of correlation between foot circumference and body length, shoulder height, and pelvic width in Sumatran elephants, or Elephas maximus sumatranus. People who are collaborating in this study are Heri Wijayanto, Denny Setiowibowo, Rini Widayanti, Tri Wahyu Pangestiningsih, Woro Danuwendo, Mahmud Aspan, myself, and also my colleague Marcel Wirabrata. This study focuses on adult Sumatran elephants, which originated from Sumatran Island in Indonesia. The weight can measure up to 2,000 to 4,000 kilograms, and it can measure up to height from 2 up to 3.2 meters. The status in IUCN currently is critically endangered. Sumatran elephant's body measurement is essential in conservation practices to perceive health status and determining medicational doses. However, due to their large size, the elephant pose a dangerous threat to mahouts who are constantly working with them. This study will help gain elephant measurements easily, therefore reducing mahouts' work risk using the correlation between forefoot and hindfoot circumference and body length, pelvic width, and shoulder height. The materials used in this study are male and female adult elephants inhabiting numerous locations in Aceh, Safari Park Indonesia, Gembira Lokal Zoo, Borobudur Park, and PLG Durip Pekanbaru. To measure the elephants, modified vernier calipers extending up to 200 centimeters and 400 centimeters, and also rollmeter were used. Measurement is carried out on body length, shoulder height, pelvic width, and foot circumference, both on hindfoot and forefoot. The results gained from body measurement were then processed to find the correlation with the foot circumference. And then, using linear regression, data were processed to gain certain formulas. Data from the formulas were then compared with real measurements. These are the measurements done to get the body measurement of the elephant. For shoulder height, it is measured perpendicularly from the ground up to the highest shoulder point. For the forefoot and hindfoot circumference, it is measured circularly on the most outer side on the distal aspect of the foot. For the body length, it is measured starting from the most front end of os frontal up to the base of the tail. And then lastly, for the pelvic width, it is measured between tuber coxae dexter and tuber coxae sinister. These are the correlation between forefoot circumference and body measurement. The correlation with shoulder height is very strong, measuring the point up to 0.80, and then for the correlation with the body length is strong, gaining the point of 0.769. And lastly, with the pelvic width is also strong, gaining the point of 0.73. These figures show a strong dispersion of the point cloud between forefoot circumference and body length, and also pelvic width, and an almost linear distribution of the point cloud between forefoot circumference and shoulder height. These are the correlation between hindfoot circumference and body measurement. The correlation with the shoulder height is strong, with the point of 0.747. And then with the body length is also strong, with the point of 0.775. And then lastly, with the pelvic width is very strong, with the point of 0.826. These figures show a strong dispersion of the point clouds between hindfoot circumference and body length, and also shoulder height, and an almost linear distribution of the point clouds between hindfoot circumference and the pelvic width. The compared data between the measured and the equation results shows that the tolerance level is all below 13%, meaning all the formulas are applicable for Mahout to measure the elephant. The more reliable formula to obtain shoulder height is using the forefoot circumference, considering the higher point of the correlation in the point of 0.809. The more reliable formula to obtain both body length and pelvic width is using the hindfoot circumference, considering the higher point of correlation of 0.775 and 0.826. This is the conclusion of our research. The most reliable formula in the result of this research are displayed as such. And the formulas gained from the research are all applicable for Mahout on the field. Therefore, the objective of the research to minimize the Mahout is achieved. These are the reference for our presentation. And now you have reached the end of our presentation. And lastly, we would like to say thank you to Universitas Gadjah Mada for providing this excellent opportunity to join the International Conference of Advanced Veterinary Science and Technologies for Sustainable Development.
Thank you very much for the presentation. So before we enter to the second presenters, I invite to the participants, to give. So this is interesting, the ladies, the girl who measures the elephant. Elephant is very big. So if there is a question for this presentation, please give your question on the chat box, And then don't forget to fill also the attender forms, which is also in the chat box. You can find it. Okay, Dr. Ma'ruf, please present for the second presentation.
Good afternoon, ladies and gentlemen. Let's introduce about myself. My name is Wayu Febriani, as a student at Faculty of Biology, Universitas Gadjah Mada. I'm from Crustacean Research Group of KSK Biogama. In this chance, I would like to present our paper about dynamics of diversity decapod in the intertidal region at Porok Beach Gunung Kidul Special Region of Yogyakarta, Indonesia. This is our abstract. Decapods were a member of tropical benthic communities and placed an important role in the coastal ecosystem. Porpoises were a center research, particularly scientific report on the level of decapod diversity. So, the research aimed to study composition structure, dynamic, level of diversity of decapod. Therefore, the study will be preliminary study for further research, the basis for biodiversity management and conservation, and the document base of marine in the Porpoise Gunung Kidul. The study was conducted in the intertidal zone at Porpoise Gunung Kidul Special Region of Yogyakarta, and then the research was carried on October 17, 2020, and January 23, 2021 during midnight. The equipment used in the research included fishnet mesh size 15 mm, ziplock, tweezer, buckets, sample bottle, universal pH, thermometer, and gasoline for station identification. The material used in the research included the clove oil, alcohol, and seawater. And then the sampling technique used is purposive random sampling, and then relaxation is done to facilitate the process of identifying sample to determine the types of species. The preliminary data of the study analyzed the diversity, the uniformity, and the dominance index of Microsoft Excel 2019. Based on the result that's shown in graph above, it can be seen that in October 17, 2020, there were 5 families found that were Diogenidae, Megenidae, Ocypodidae, and Centenidae. While in January 23, 2021, the diversity of crustaceans was found is more higher. There were 7 families found consist of from Diogenidae, Epinidae, Clepsydae, Megidae, Ocypodidae, Protinidae, and Centenidae. From the 2 sampling that was conducted in the most species found came from family Protinidae. Table 1 is species diversity of decapod found at Porpoise Gunung Kidul Special Region of Yogyakarta, Indonesia. Meanwhile, it can be seen that the count of individual at Porpoise in January 2021 was higher than the count of individual at Porpoise in October 2020. Family Protinidae shows in the highest count of individual in January 23, 2021, which was 9 individuals. Likewise, on October 17, 2020, the family Protinidae showed the highest individual count, which was 4 individuals. Stability of water decapods can be described from the diversity index, uniformity index, and dominant index. The value of diversity, uniformity, and dominance of decapod on the beach in post-sampling time difference is shown in Figure 4. Based on the result of the study obtained that Shannon winner with diversity index value 2.3. The highest value of decapods diversity index and low in the intertidal zone of Porpoise is supported by rarely visited by tourists and the state of Porpoise substrate. Uniformity index value 0.05 to 0.1. The uniformity index value was close to 0, which indicates that the level of uniformity is low. The dominant index value belongs to the low category. The condominance index value was close to 0, so the result shown that there are no other species or decapod community structure on Porpoise relatively stable. The dominance of certain species in a water may indicate the presence of ecological pressure of the seawaters. Therefore, based on the result of the study, it is known that the level of dominance is low, so that there is an ecological balance. The data presented in Table 2 were environmental parameters in the intertidal region at Porok Beach. Based on the research, the environmental parameters at Porok Beach that have been measured affect, among other, water temperature and water pH. One of the factors that affect the survival of organisms in the ocean, especially decoupled, was temperature. Temperature plays a role in the physiological and respiration process of living things, so that it affects the metabolic process in organisms. The result of the research on pH at products were 7, which were classified and neutral, because the pH of the seawater was neutral and very good for the reason of decoupled organisms and waves. The rainy season with high waves is found on October. Meanwhile, January was still the rainy season, but the waves are not high as in October. The heights of the days wave can affect the excitement of individual decouples. Based on research conducted, it was concluded that family Dioclinidae, Epinidae, Clepsydae, Maginidae, Ocypodidae, Potonidae, and Centinidae as the composition structure of decoupled on porok beds. Decoupled dynamics as of January 23rd, 2021 are higher than October 17, 2020. Shannon-Wiener Pitts Diversity Index value 2.3 and Uniformity Index value 0.05 to 0.1 and Domination Index value 0.002 to 0.1. Water temperature, season, waves, and the number of visitors limiting factor that affect the dynamics of decoupled at porok beach.
Okay, thank you very much for the first presentation. And then we will going to the second presenters. For the participant, don't forget to fill the attendance form of this class, yeah? Dr. Mahfud, please present for the second presenters. Oh, we are already finished, yeah?
Yes.
Sorry. I feel 3. We are done. Okay, so I will have a look. Okay, seems there is no question. So if there is no question, so I will ask, yeah. So don't worry, my question is not difficult, yeah. The first one, it's this is I just want to we share and discussion, yeah. The first one for the 11. So this is a very nice work, yeah. And then do the formulation to measure the foot circumference and then also body length and pelvis width and others. And then is it the sample is Elephas maximus sumatranus. Is the formula we can use to the other species of elephants or only on this species? And then maybe you can also tell us in common what is the advantages to the morphometric study for the 11.
Okay, for Dr. Happy, thank you very much for the question. I will try to answer. Firstly, for the research that we did, we found the formula exclusively only for the Sumatran elephant. So it is not applicable on other elephants because the morphometry anatomically with other elephants are different. And secondly, the main purpose for this research is to gain accurate numbers for the body measurement without having to measure it manually, because it is very hard to get perfect measurement due to the large size of the elephant. And then also, it is also for minimizing the work risk for the Mahfuz.
Is the body measurement usually done for the 11 to maybe we will, so with the body measurement we will need to know the information about the health status, something like that. It's common do in the quarantine for the 11 or something like that.
Yes, it will. Yes, it will be very helpful to get to do the recording for the elephant, and also later on if we need doses for medication and so on and so forth, and also it can be used to predict the number of the population. So by measuring the foot circumference, we will know how many male or female or even the cups that one population has.
Okay, it's very interesting research, yeah, and also good luck for the other research. Maybe use another 11s, yeah, another species. I mean, and then I will have a look in the chat box. Okay, I think there is still no question, so I ask again. Okay, Miss Wahyu, I have maybe this one. This is last question before we close our very interesting and warm parallel class. Yeah, why actually? Actually, what is the definition? Because I'm not a biologist, yeah. This is my question. It's just interesting. Your research done in the midnight to collect the decapods, yeah, and then you use the intertidal zones. Actually, what is the intertidal zones, and why you choose intertidal zone in Porok Beach as an area for your samples of decapods? Please.
Okay. We choose intertidal zone because the intertidal zone tends to be enveloped by algae in wave-sheltered conditions and by snail, pinnacle, and shellfish, as was explored in grass. Relative soft rock such as limestone and limestone can support living species.
Okay. So you go to the Porok Beach at midnight, yeah?
Yeah, yes.
But with your friend, of course, yeah?
Yes, with my friends in my group research ICAVESS Biogama.
Okay, it's I think it's very cool, yeah, midnight going, and then you take directly, and they come easily in the
sorry, can you repeat?
So you come midnight going to the beach, and then what you do so you can find the decapods? They are come easily on the surface, or you should have look.
Apa itu namanya?
You catch.
Langsung
directly catch.
Directly catch gitu. When midnight we directly catch gitu.
So maybe you choose the midnight because in the midnight the decapods going to the surface or something like that, yeah. They are going more easily in the surface.
Yes.
Oh, okay, okay, okay. Interesting. Two ladies with the 11 and midnight going to the beach. Maybe Dr. Andre, you can go midnight to the beach swimming, yeah. Okay. Maybe this is the last session for our parallel session in the class B. Maybe it's so tired, yeah, but this is very interesting. There is so many questions, and Dr. Andre will give a greeting close. So time for you, Dr. Andre. Thank you very much, and applause, yeah, for all of you.
Okay, thank you for the last session presentator. Okay, I think we cover up everything, right? All presentators came to these sessions, and I think I'm gonna sum up what we've been doing this afternoon. So it was a fruitful discussion, all of the session, from wild to pet animal, from small to big size, and I think we have a diverse presenter here, from veterinarian and from the biology and the forestry I saw just now, and we all here only to build a better world for a sustainable development for our future ahead. So I hope it will be beneficial for all of us and for our knowledge. And personally, I learned so much from this realization, because yeah, so many insight like from the biological side. And I think that's all from my side. Miss Dr. Hefi, do you have anything to add before we move on to the like final session?
For me, I just want to say thank you. This is a very interesting class, yeah. In great honor for us to know that we have a very big biodiversity, yeah. And then we learned so many things that Dr. Andre already told to us. Hopefully it's keep us spirit to work in our biodiversity. If not us, who will do? Okay. And thank you very much for your participation. Maybe Dr. Ande will guiding us to take a picture or something like that before we are leaving.
Before like any other conferences, like taking pictures, I would like to remind you to fill the attendees form to get your certificate at the end of the event. And you should also attend all plenary session and also the parallel session. And we have, I think, 10 room for the parallel session, and you can choose which room that you're interested in by looking at your guideline book. I think we already shared the guideline book in our WhatsApp group. You can check, and you can, I think, choose which room that you think might fit into your like curiosity. And for tomorrow's session, we will have a plenary session at 8 in the morning. So I hope we can see you there. Last but not least, group picture. So I'd like to invite all of the participants here to turn on their camera, the co-host, the presenter, and the participant. Please turn on your camera so that we can take a picture. And yeah, I think it's a precious moment here. The first international conference on advanced veterinary science and technology for sustainable development. Okay, let me see. Are we all okay? Okay, I think we will wait for another 10 seconds. Wait, okay. For our co-host, is it enough, or do we still need to wait for the others to turn on their camera?
We need to still wait.
Okay.
For several participants.
All right. I think there are 1, 2, 3, 4, 5, 8, 11. I think around 15. Okay. 12. Okay. I think the majority of us already turned on their camera.
I think we can proceed, Dr. Andre.
Okay. Okay. On the count of 3, please smile, and our co-host will take a screenshot of this Zoom meeting. Okay. 1, 2, 3. Maybe again, Miss Wahyu is like kind of moving there. Wahyu Febriani. Okay. Start again. 1, 2, 3. Other posts maybe. ICAVESS. I don't know if ICAVESS has posts, but ICAVESS. Okay. Do we need more picture, Miss Obari or Dr. Mahru?
Yeah, I think once more because we are 2 page.
I see.
So yeah, I'm sorry.
Okay, no worries. And I think more people are coming. Okay. Again, just smile, guys.
I will count 1, 2, 3. Once more. One, two, three. Yep, we're done.
Okay.
So we are done for today, Dr. Andre.
Yes.
And then thank you very much, and we see you in next year, maybe in the second ICAVESS, hopefully.
Hopefully. Well, we still have tomorrow before next year.
Yeah.
So see you tomorrow.
See you tomorrow.
And it's been a pleasure to moderate this parallel session, and stay safe and
stay healthy. Wait for the vaccination soon, yeah?
Yeah. Have it first, like
today.
I'm the last
As
of the
yesterday.
priority here.
Mhm.
I'm the last of the priority here. Just
Okay. Bye-bye.
everyone. Thank you.
Thank
Thank you,
you.
Dr. Andre and Dr. Happy. Thank you to all participants.
Thank you. Everybody allowed to leave this room meeting. Masih ada. Oh. Dok Andre,
Ya, Dok.
udah inform Atika belum ya buat besok bagaimana? Saya belum ada komunikasi sama apa sama dia sih.
Oh, belum. Ya, ntar saya coba komunikasikan, Dok.
Ya, atau aku soalnya
Kayak
belum.
gini kan, Dok? Kayak
Ya,
gini kan?
gini aja,
Gini
santai aja. Enggak. Aku ya kita bisa sama apa ya spontan juga nggak masalah sih. Nah, tapi masalah sebenarnya enakan ya kalau pasangan ini besoknya nggak usah diganti. Soalnya udah tahu ya Dok Andrea.
Iya, Dok. Ya gimana, Dok? Pemberian awal ya.
Mohon maaf, saya pembagian tugas saya di sini apa PJ PIC video jadi
Iya, kan kalau misalnya kemarin kan Gladys sudah sama Dok Andrea, kita udah chatting udah apa kan sebenarnya chemistry-nya udah dapet tuh ya Dok ya. Jadi ya oke lah. Good luck ya buat besok. Saya izin itu dokumentasi nanti yang bikin siapa? Dok Waris sama Dok Ma'ruf ya?
Dr. Waris sama ada ini mahasiswa S1 ini Mbak Rara.
Oke.
Yang
makasih.
co-host itu.
Makasih banyak ya.
Ini
Ya sama-sama Dok.
Kita udah nggak ada tanggungan pertanyaan ya, semua dan ya.
Iya sudah sama yang jelas jangan lupa untuk isi best presenter ya. Dok.
Ya aku belum isi soalnya, aku nunggu hari ini sih kalau aku maksudnya apa ya kan beda ya rasanya ya. Ya udah ntar habis ini aku isi. Ya. Makasih banyak ya semuanya saya izin.
Ya terima kasih banyak. Happy sehat-sehat selalu.
Sehat. Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh.